Good afternoon, everyone. I'm Dr. Shomi Bhattacharya. I'm a chemistry faculty in Extramarks. Today, I will be presenting the chemistry solutions of AIRTS Mega Test 1. But before that, let's talk about the test series. So basically, we have three ongoing test series, that is class 11th, 12th, and AIRTS. So AIRTS has started from 28th November 2022, and it caters classes 12th and 12th plus. So it will be covering the subjects, all the four subjects, that is physics, chemistry, botany and zoology. Basically, the number of tests, there are four minor tests, two half syllabus tests, and 6 full syllabus tests. So basically, So basically, it will be based on the NEET pattern. There will be 200 questions, and it will be for 70 maximum marks. So the duration of the test series will also be from 3 hours, 20 minutes. All right. So today, we will be taking up the Mega Test 1, the solutions of Mega Test 1, which was conducted on 23rd January. So the first four, minor test 1, minor test 2, test 3, and test 4 have already been conducted. So let me just take you through the chemistry solutions of the Mega Test 1. All right. So let's start with the first question. The first question, in the first question, the element of the highest metallic character has been asked. Okay, so there were four options: nitrogen, phosphorus, antimony, and bismuth. So let's talk about the concept. So generally, down the group, electronegativity decreases. So if electronegativity decreases, then electropositivity will increase. So if electropositivity increases, then the metallic character will increase. So if that is the case, herein we are taking group 15 elements, right? Since we are taking group 15 elements, so over here, bismuth will be the one which has the highest metallic character. So bismuth will be the correct answer, that is D. Moving ahead, let me take the question number 2. In this question, they have asked in which one of the following arrangements the given sequence is not strictly according to the properties indicated against it. So as you can see, the A, B, and C options are correct, but in the D option, they have given in accordance with the increasing pKa values, right? So which is actually not true. So if you move down the group, the bond dissociation enthalpy decreases. So if that is the case, this is the order of increasing acidic strength. If this is the order of increasing acidic strength, then the Ka value will increase and the pK value will decrease. So D is the incorrect arrangement. Now let me take the question number 3. Here the hydrolysis of IF5 has been asked. So it's based on P block elements. It's a basic reaction based question where IF5, if it undergoes hydrolysis, it is going to give you HiO3 plus HF. So if that is the case, the correct answer has to be C. All right. Now moving ahead, let me take the question number 4. Here, which one of the following pair of compound is isoelectronic and isostructural has been asked. Now if I'm talking about isoelectronic, what does isoelectronic mean? Isoelectronic means it will have same number of electrons. And if I'm talking about isostructural, this means it will have same structure. Now if that is the case, if you look into the options, all the option has XeF2. So let us just find out the number of electrons and the structure of XeF2. So XeF2 has how many number of valence electrons? That is 8 plus 7 plus 7. So if you calculate, you're going to get 22 number of electrons. All right. And it also has a linear structure. So basically the electron pair number over here is, if you don't know how the linear structure came, so the electron pair number it will have is 5. So it has bond pair. Basically how many fluorine has bonded with the xenon? There are 2 fluorine atoms, so 2 bond pair, lone pair 3, which makes the structure linear. So we are actually looking for this 22 linear combination. Now this matches with the C option where I3 minus is given. So I3 minus can also be written as I2 minus. Alright. So if that is the case, let's just count the number of valence electrons. How many valence electrons will this have? 7 plus 7 plus 7. And since there's a minus 1, so this is plus 1. So this gives us a total of 22 electrons. And if you calculate the electron pair over here, the electron pair will come out to be 5. If the electron pair is 5, okay, if you calculate it in the similar manner, that will be half plus 7 plus 2 plus 1, you're going to get 5. And here also the bond pair will be 2, lone pair will be 3, which gives us a combination of linear. So this tallies with the C option. So C is the correct answer. Moving ahead, let's talk about question number 5. Here ammonium dichromate undergoes decomposition to give you A plus B plus C. And this is a very simple reaction based question. So here the correct answer is C, which gives us nitrogen, Cr2O3 and H2O. Moving ahead, let me take the next question. Question number 6 is based on the oxo acid which has PP linkage. Now when I'm talking about PP linkage, there are 4 options: hypophosphorous acid, orthophosphoric acid, hypophosphoric acid and pyrophosphoric acid. Now let's look at the structures. So these are the structures, and from here it is very clear that hypophosphoric acid has a PP linkage. So what is the correct answer in here? The correct answer is hypophosphoric acid. Let's move to the 6th question. Let's move to the 7th question. I'm so sorry. 7th question has 2 statements. The statement number 1, here the vulcanization of rubber has been catered, and this, in vulcanization of rubber, the sulfur crosslinks are introduced. Okay, this is the statement 1. And the statement 2 has vulcanization is a free radical initiated chain reaction. So we need to answer which of the following, whether statement 1 is true, statement 2 is true. So it's a statement based problem. So let's just understand what exactly is vulcanization. So in vulcanization, the natural rubber is treated with sulfur or compounds of sulfur under steam or at extreme pressure to give us polyisopropene chain with sulfur bridges. So this is the vulcanization process. With the help of vulcanization, what happens? The mechanical strength of the rubber is increased. So now statement 1 is correct because in vulcanization of rubber, the sulfur crosslinks are introduced. Plus, vulcanization is a free radical initiated chain reaction. This is also correct. But as you can see, the statement 2 is true, but it is not a correct explanation for statement 1. So B is the correct answer. Moving ahead, let's talk about the question number 8. Here the monomeric unit of nylon 6 has been asked. As you can see, there is a confusion about nylon 6,6 and nylon 6. So basically, as you know, nylon 6,6 has 2 monomers, alright? That is adipic acid and hexamethylene diamine. But when we are talking about nylon 6, it has only 1 monomer, that is caprolactam. So what is the correct answer? The correct answer over here is B, that is caprolactam. Alright, now moving ahead to the next question, that is question number 9. The question number 9 is from Chemistry in Everyday Life chapter. Now here they have mentioned the non-narcotic analgesic out of the following. First of all, what is non-narcotic analgesic? This means this is non-addictive pain reliever. Alright, so the first 2 options are tranquilizers, while penicillin is an antibiotic. So the correct option over here is aspirin, that is C. Alright, now moving ahead, let me go to question number 10. Here, this question is also based on chemistry in everyday life. Here they have asked the compound which is an artificial sweetening agent over here. Saccharin, sucralose, aspartame, and all of these has been mentioned over here. D, that is all of these, saccharin, sucralose, aspartame, all of them are artificial sweetening agents. So all of them are correct. So the correct answer is D. Moving ahead, now this question is based on chemical kinetics. Alright, now let's try to understand the question. Over here, in a reaction, A is giving rise to products when start is made from 32 into 10 to the power of minus 2 molar of A has been mentioned. Okay, so this means one concentration, say A1, is mentioned as 32 times 10 to the power of minus 2 molar. The half-life is given as 240 minutes. Alright, in the next part, for the initial concentration, that is 16 times 10 to the power of minus 2, this means this is A2, 16 times 10 to the power of minus 2, the half-life mentioned is 480 minutes. Now you have to calculate the order of the reaction. So let's try to solve this question. So over here, we will be using the formula T half 1 divided by T half 2, that will be equal to A2 by A1 to the power of n minus 1, where n is the order of the reaction. Now let's substitute the values of A1, A2, T half 1, T half 2. Alright, so over here we can just write, let me just substitute all the values. So 240 by 480, this should give us A2 value over here is, as you can see, 16. So 16 times 10 to the power of minus 2 divided by 32 times 10 to the power of minus 2 to the power of n minus 1. So let's try to calculate. So we are going to get 1 by 2 will be equal to 1 by 2 to the power of n minus 1. So n minus 1 is equal to 1. So n is equal to 2. So what is the correct answer? The correct answer over here is C, that is 2. So this is the second order reaction. Alright, moving ahead, let me take the question number 12. Here you have to select the graph that doesn't belong to first order reaction. Over here, the first, second and the third graphs are correct because in the first graph, the T half is independent of A naught, that is correct. It's a first order reaction, that is what happens. Even the B part and the C part are correct. The only incorrect answer is D, that is this curve. Basically, the exponential equation of the first order reaction is this. This doesn't tally with this particular equation, that is why D is the incorrect, incorrect graph which doesn't belong to the first order reaction. Moving ahead, let me take the next question. This is also based on kinetics. Now for a first order reaction, the time required for 99.9% completion has been asked. Okay, so what is the formula? For first order reaction, T is given by 2.303 by K into log of initial concentration divided by the final concentration. Over here, if A is equal to 100, then we are talking about 99.9% completion. So X will be 99.9. Now substituting all the value and let's try to solve the question. So T will be equal to 2.303 by K log of 100 A, 100 minus 99.9. So this is going to give you 2.303 by K log of 100 by 0.1. So if you solve it, you're going to get 2.303 by K times, since log 10 to the power of 3 is what you're going to get, so times 3 will be the correct answer. So over here, this answer tallies with the C option. So C is the correct answer. Moving ahead, let me take the question number 14. Now over here, I will try to solve the question using a trick. So let's try to solve it. Over here, the time required for 87.5% completion is 300 seconds, and you need to calculate the time required for 93.75% completion. How do we solve it? So we have a trick for it. Generally, if T half is the half-life period, then T 87.5% is given by 3 T half, and T 93.75% is given by 4 T half. So from here, from this particular equation, we'll try to calculate the half-life period. So let's try to calculate the half-life period. The half-life period comes out to be 100 seconds. Now we'll just substitute this value in here. So what is the value that we get? 400 seconds. Alright. So the correct answer is C, 400. Alright. Moving ahead, let me take the next question. The next question is based on metallurgy. Now here, consider the following statements and you have to choose the correct options. As you can see, we are talking about aluminothermite process, and here aluminium acts as a reducing agent. So if aluminothermite process have been asked, then Fe2O3 plus aluminium should give you Al2O3 plus iron. Alright, if this is the equation, definitely over here aluminium is acting as a reducing agent. Again, the process of extraction of gold involves the formation of AuCN twice minus complex and zinc tetracyano complex, which is correct. Even in the extractive metallurgy of zinc, the partial fusion of zinc with coke is definitely called sintering, and the reduction of this ore to molten metal is called smelting. So this is also a correct option. Extractive metallurgy of silver from its ore argentine involves the complex formation and displacement by more electropositive metal. Now we have to choose the correct option. As you can see, A, B, C, D, even D is also correct. All of these options are correct. So D is the correct answer. Moving ahead, let's take the 16th question. Now here, this question is based on match the following. So over here, the whole concept is based on the processes taking place in metallurgy. So as you can see, the cyanide process, they have asked, we just have to match it. So cyanide process is an extraction of gold, we have just seen in the previous statement. Froth flotation will be involving the dressing of zinc sulfide. Electrolytic reduction will be extraction of aluminium because aluminium is very reactive, and zone refining will include ultra pure germanium. So basically A is the correct answer. Alright. Now moving ahead, let us take question number 17. Here the total number of optically active isomers of 2,3-dibromopentane has been asked. Okay, so let's try to draw the structure. So this is 2,3-dibromopentane, and as you can see, there are two chiral centers, this and this, plus there is no symmetry element attached. So the formula that we will be using is 2 raised to the power of n, where n is the number of stereo centers. So that is 2 raised to the power of 2, that is 4 answer. So the correct answer over here will be 4. Alright. Moving ahead, let me take question number 18. Now this is based on solid state. Now here, how many unit cells have been asked that can be made out of 18 carat pure 197 gram sample of gold, and here gold has an FCC structure and the atomic mass of gold has been mentioned. Alright, so let's try to solve this question. So what do we have to calculate? we have to basically calculate the number of unit cells. Okay, so we have to calculate the number of unit cells. What else information do we have? We have FCC structure, so the value of Z is equal to 4 for an FCC structure. Alright, so let's try to solve. The first thing that we need is the mass of pure gold. Otherwise, how will we calculate the number of moles? So if we are talking about 18 karat gold, this means it has 18 part of pure gold and 6 part of other metal. So the mass will be given by 18 divided by 24 times 197. So if you solve it, you are going to get 147.75 gram. Now the second part, in the second part we need to calculate the number of moles. So number of moles is given mass upon molar mass. Given mass will be 147.75 divided by the atomic mass, that is 197. You get a value of 0.75. In the next step, we have to calculate the number of gold particles. To calculate the number of gold particles, we'll use our formula, number of moles times the Avogadro's number. Okay, so number of moles is 0.75 times the Avogadro's number. Okay, in the last step, we will be calculating the number of unit cell. The number of unit cell will be equal to 0.75 divided by 4 times the Avogadro's number. So if you solve it, you're going to get 3 times Avogadro's number divided by 16. So A is the correct answer. Now, moving ahead, let me take the question number 19. Now, this is also based on solid state. Here, the ionic solid XY has a BCC structure with an edge length of 4.4 angstrom. They have asked about the minimum distance between two oppositely charged ions. So the options are given A, B, C, D, and we have to calculate the minimum distance. Over here, we have to use the formula. Root 3A will be equal to 2 times the minimum distance between the two oppositely charged ions. So let's just calculate the minimum distance. That will be root 3A by 2, that is 1.73. What is the edge length? 4.4 divided by 2, and if you solve it, you're going to get 3.8 angstrom as the correct answer. Let's move to question number 20. This question is based on coordination chemistry. This is a very simple, very scoring question. Here the coordination entity which absorbed lowest wavelength of light has been asked. Now when we are talking about lowest wavelength of light, what concept should we use? So if we are talking about lowest wavelength of light, wavelength will be inversely related to the stability of the complex. So over here we just have to find the least stable complex. Over here the least stable complex, what are the conditions? The weak field ligand should be there, and the if you are talking about greater the stability, so it should have a strong field ligand. Over here the strongest ligand is Cn. Okay, and the oxidation number of the central atom should be the highest. So this actually tallies with only the A option, where cobalt is in the +3 oxidation state and Cn is a strong field ligand. So this will have the lowest wavelength of light. Moving ahead, let me take question number 21. Here the magnetic moment of scandium plus X, okay, is 0. Now what does this mean? This means that the magnetic moment is 0. This means the number of unpaired electron is 0. Now here they have asked the oxidation state of scandium. That's why they have mentioned scandium plus X. How will we do it? So scandium generally has the electronic configuration of 3d1 4s2. So if you are removing 3 electrons from the scandium metal, you're going to get 0 unpaired electron. So scandium plus 3 will have the electronic configuration of this, and this will have 0 unpaired electrons. So the correct answer will be 3. That is C. All right, now moving ahead, let's go to question number 22. This is based on IUPAC number. Here the potassium dicyanobisoxalato nickelate 2 has been given, and you have to tick on the correct option. How do we do it? We have potassium, we have dicyano, we have dioxalato, and we have a nickel. So it's a anionic complex. Over here, let's just find what charge shall be put. Nickel has the oxidation state of 2. Cn will have -1 charge, so this will cater as -2. Oxalate ion has -2 charge, so there are 2 oxalate ions, so -4 will be there. Now to neutralize this, we need to put a 4 in here, right? So the correct answer over here will be A, that is K4 NiCN twice, oxalate twice. Moving ahead, let's take question number 23. This is also based on coordination compounds. Here the outer orbital complex has been asked. As you know, whenever we are talking about outer orbital complex, obviously strong field ligand will not be catered, so C cannot be the answer. It has to be A, B or D. Now, if you look into the options very clearly, here the copper will have basically D9 configuration. So the chances of this complex having an outer orbital complex will be very, very high. So the correct answer will be D. All right. Moving ahead, let me take the next question. This is on crystal field stabilization energy. That will be minimum in. So what are you searching for? So if we are talking about minimum, so if we are talking about say minimum crystal field stabilization energy, so we need to look for smaller oxidation number on the central atom. That should be the thing, and we are also looking for a weak field ligand. So this tallies with only one option, that is C. So over here, the ligand is also weak field, and cobalt has the least oxidation state of +2. So the correct answer will be C. All right. Now moving ahead, let me take question number 25. Here you have to select the complex which has maximum number of isomers. It's super simple. You can see A, B, C will hardly have any different types of isomers because of its symmetrical structure. It's only C option. Okay, A, B, D will hardly have any isomers because of its symmetrical structure. So it has to be C, and these are the isomers which will be possible. All right, moving ahead, let me take question number 26. Here the N factor of potassium iodide when treated with acidified KMnO4 has been asked. So this is the equation. As you can see, when we are converting MnO4 minus to Mn2 plus, 5 electrons are required. But if we are converting iodide to iodine, only 1 electron is required. All right, so the correct answer over here will be 1. All right. Now moving ahead, let me take the next question, that is question number 27. Ions which does not have 3d6 configuration has been asked. All right, so over here, cobalt 3 plus, nickel 4 plus and Fe2 plus is given. Now this tallies with only C, where the electronic configuration will be, as you know, the outer electronic configuration, outer electronic configuration of chromium is 3d5 4s1. Now if you are talking about chromium plus, the electronic configuration will be 3d5, which is not tallying with 3d6 configuration. The rest of them will have 3d6 configuration, so C is the correct answer. Moving ahead, now this question is based on DNF blocks. Here we are talking about the incorrect statement about lanthanoids. Let's go through the statement. All lanthanoids are silvery white soft metals. It's correct. Many trivalent lanthanoid ions are colored. This is also correct. But these are bad conductors of heat and electricity, which is not correct. Okay, but still let's just go through the fourth statement as well. Hardness increases with increase in atomic number. This is also a correct statement. So C is the incorrect statement, where it has been mentioned that lanthanoids are bad conductors of heat and electricity. Moving ahead, let me take the next question. The next question is on electrochemistry. So over here, if the E0 cell is greater than 0, you have to select the correct relation. So if the E0 cell is greater than 0, the correct relation will include delta G0 value less than 0 in order to have a spontaneous reaction. So if E0 cell is greater than 0, then the reaction is considered as spontaneous. And for a spontaneous reaction, the free energy change should be negative, that is less than 0. So what is the correct answer in here? The correct answer in here will be C. Alright, moving ahead, let's take the next question. This is based on the Colroche law. Here, they haven't asked you to calculate anything. It's a super simple question where the molar conductance of Na2SO4 at infinite dilution has been asked. So since we are talking about Na2SO4, so the correct answer, it will give you 2Na plus and SO4 2 minus. So this will be matching with the B answer, where we take the 2 times the ionic conductivity of Na plus plus the ionic conductivity of sulfate. So B is the correct answer. Moving ahead, let me take the question number 31. Here, this is based on the Nernst equation, which is super simple to do. Over here, the EMF of the cell at 25 degree will be for this particular cell they have given. Okay, so for this particular cell, let's try to calculate the, it's a concentration cell, right? So E0 value they have given 0.80, which is null and void. So here we will be using, this is the Nernst equation. Now if you look into this particular cell, the correct cell equation will be, so basically if you don't know how to write it, so first let me just show you how to do it. So first, silver will give you silver plus electron at anode, and at cathode silver plus electron will give you silver. Now here we are talking about 0.01 molar. Here we are talking about 0.1 molar, right? So this will give us this particular now, in case you have no idea, that's why I just gave you an insight about how I will be just catering the concentrations. Now let me just use the Nernst equation. So here E0 cell value will be equal to 0. So E cell will be equal to minus 0.0591, since n is equal to 1 because only one electron was transferred, log of, okay, so we will be first taking 0.01 divided by 0.1. So if you solve this, you're going to get 0.0591 as the answer, which tallies with the A option. Moving ahead, let me take the 32nd question. Now this is based on solutions. A solution containing 36 gram per decimeter cube of glucose is isotonic. Okay, this means they have same osmotic pressure with 3% solution of non-volatile and non-electrolyte solute, and we need to calculate the molar mass of the solute. So let's try to do it. So over here, basically, the two osmotic pressures are equal, right? This means C1RT will be equal to C2RT. Now, if you have to convert the concentration into mole per decimeter cube, what do? You will be taking the given mass, that is 36, divided by the molar mass, that is 180, And since we are talking about per decimeter cube, so the volume is also 1. RT gets cancelled, which I forgot to do. And in C2, we will be taking, since they have given 3% weight by volume, this means 3 gram is the given mass. Let's consider the molar mass to be X, okay, times the volume. Over here, if we talk about the volume, okay, so volume will be 100, volume of the solution will be 100 ml. So let's divide it by 1000. This should be the case. All right. So if you solve this, you're going to get 150 gram per mole as the answer. So what is the correct answer? The correct answer over here is 150 gram per mole. Moving ahead, let me take the question number 33. Now over here, which one of the following aqueous solution has highest vapor pressure? To have highest vapor pressure, what do you need? To have highest vapor pressure, the concentration of the solute should be, what do you think? It should be low, okay? And the temperature should be high. So this actually tallies with the D option. This actually tallies with the D option, where the concentration of the solute is just 0.005 molar, which is one of the lowest, and the temperature is high. So if the temperature is high, the vapor pressure will also be high. So D is the correct answer. Moving ahead, let me take the 34th question. Here, soap lather is an example of, this is very simple, soap lather is an example of foam. Moving ahead, the next question is also based on surface chemistry. Here the incorrect statement for chemis option has been asked. Now generally chemis option is usually irreversible in nature. That's correct. It's an exothermic process. That's also correct. It decreases with increase in surface area of the adsorbent, which is not correct. It increases with increase in surface area of the adsorbent. So C is the incorrect statement, and it is highly specific in nature. That is also correct. So C is the incorrect statement. Moving ahead, let me take the 36th question. Now this is based on biomolecules chapter. Here the water soluble vitamin has been asked. Vitamin A, D, C and E has been asked. So the correct answer over here is C, that is vitamin C. Moving ahead, now this is also based on disaccharide portion of biomolecules. Here you can see the sugar molecule formed by the linkage of C1 of beta D galactose units and C4 beta D glucose unit is. Okay, let's go through the structure. This is sucrose, this is starch, maltose, lactose. It is very clear. The correct answer is lactose, where C1 beta galactose is linked with C4 of beta D glucose. So the correct answer is D. Let's take the 38th question. Now this is from here onwards, we will be discussing the proper organic part. So over here, the C5 compound A has C5H10Cl2. This on hydrolysis is giving us B, that is C5H10O. Now this reacts with NH2OH. This means it can form an oxime or hydroxylamine. Okay, so basically it's we are treating it with hydroxylamine and it will form an oxime. Other than that, it also gives iodoform test, positive iodoform test. Okay, and in the question they have also said it does not give Felling's test. This means it is giving negative Felling's test. This means we are talking about a ketone. Okay, so we are talking about a ketone which has CH3CO group because it is giving you positive iodoform test. Alright, so what will be the correct answer? So C5H10O is mentioned where CH3C double bond O group is present. So obviously if you subtract C2H3O from C5H10O, you're going to get C3H7. So let's just put CH2, CH2, CH3. So this has to be our ketone. Okay. Now this basically we are getting This is basically obtained from a geminal dihalide. So when we are talking about geminal dihalide, so there are A, B, C. As you can see, A and B over here are A, B and C are geminal dihalide, where the halogens are present on the same carbon atom, while D is a vicinal dihalide, so D cannot be the answer. So it has to be A, B, C. So let's see which is the correct answer. So it is possible only if 2Cl are present on the second carbon where the carbonyl group is present. Now, this is the correct answer. All right. So, which tallies with the A option. Now, if you're wondering how did I understand it gives a positive iodoform test, as you know, iodoform reaction is that reaction which is given by those carbonyl compounds which has a CH3CO groups. So over here, what is the basic reaction? If you don't remember what is iodoform reaction, so basically, so if you have a CH3, say you have a CH3CO group and you have an R group, and if you're treating it with I2 and a base, any base you can take, so what do you get? You get RCO gets oxidized to RCONA, and this CH3 gets converted to CHI3, which is our iodoform. Alright, so from here this group was confirmed, and since it gives a negative Felling's test, this means since it gives a negative Felling's test, this means aldehydes are not present, it is only ketone. Alright, now moving ahead, the next question is based on the increasing acidity. Now over here, as you can see, there is an orthobenzoic acid present. From the ortho rule, basically you are very much aware that an ortho substituted benzoic acid, no matter what substituent is there, will always be highly acidic. So over here, basically ortho nitrobenzoic acid will be highly acidic, and the para-hydroxybenzoic acid will be the least acidic. Alright, so as you know that acidic character will be directly related to minus I or minus M effect, minus I or minus M, and inversely related to plus I or any group which shows minus I or minus M effect, and will be inversely related to any group which shows plus I and plus M effect. So over here, as you can see, NO2 will be showing the minus I minus M effect. So basically, one will have highest acidic character, followed by 2. Okay, then we'll have 4 and then 3. This tallies with the D option. Alright, moving ahead, let me take the 40th question. The 40th question is based on an ABC type reaction based question. Now over here, the first reaction where benzene is treated with CO plus HCl followed by the catalyst N-hydrous AlCl3 or CuCl is nothing but catamaran aldehyde synthesis. Over here, what do you get? You basically get from benzene, benzaldehyde will be obtained. Okay. Now, if you treat it with K2Cr2O7, K2Cr2O7 is an oxidizing agent which is going to give you benzoic acid. Followed by it, when you're treating an acid and alcohol, if you treat an acid and alcohol in the presence of conc H2SO4, you're going to get an ester. So what do we get if you treat it with C2H5OH followed by H2SO4? You're going to get an ester. What is the ester like? COO, C2H5. Okay, so C over here basically is an ester, so B is the correct answer. Moving ahead. Now the next question is based on reagents. Okay, so the above conversion can be carried out by which one of the following reactions? Let's look at the carbon number. Over here, if you just exclude R, you can see on the right hand side there are 2 carbon atoms attached to the alcohol. In the left hand side there are 2 carbon atoms attached to the OH, and in the right hand side there are 3 carbon atoms. So we are increasing the carbon chain. This means CN has to be introduced. How do we do that? Okay, so what do we do over here? we will be converting alcohol OH into Br like so. Followed by it, we will be treating it with KCN, not silver cyanide, because silver cyanide will give you isocyanide. Now this Br will be converted to Cn. Followed by it, we'll underwent hydrolysis. What do we get? We get our desired product. So what should be the correct answer over here? So the correct answer has to be C. Alright, moving ahead, let's go to question number 42. Over here, you can see we have cyclohexanol. It is treated with PCC. So what is it going to do? So if you treat cyclohexanol with PCC, which is a mild oxidizing agent, you're going to get a carbonyl group, that is ketone. From secondary alcohol, you get a ketone. Followed by it, you're treating it with zinc amalgam HCl. What is this reaction called? This reaction is called Clemenson's reduction, where C double bond O will be converted to carbonyl group will be converted to an alkene. Alright, so what is the correct answer in here? D. So cyclohexane will be the correct answer. Moving ahead, the question number 43. Over here, metanitroaniline has been taken and it is treated with Br2 acetic acid, and they have asked you the major product. Generally, NH2 over here is a powerful activator, but over here you can see that this position and this position will have lot of blockage. Basically ortho will basically over here, if we substitute in this particular ortho position, there will be a problem. Again, if you substitute in this particular position, again there will be a problem. So the only option we have is this position. Why? NH2 is an orthopara directing group. Where will it direct the Br? So it will direct the Br, that is Br plus, which is the electrophile over here, to the ortho position in here. Alright. Now let's take question number 44, the reaction which gives propylamine as the major product. Now over here we are trying to synthesize propylamine. Propylamine will have 3 carbon atoms attached to the NH2. So as you can see, the first one has Hoffman bromamide's reagent. So obviously that won't be possible because over here the CO will just leave and we are going to get 1 carbon less, so we'll get ethylamine. This is not possible again because it will just convert the amide into nitrile, and then we have C where a nitrile is present. So this nitrile on reduction will definitely give us CH3, CH2, CH2, NH2. Okay, so this is the correct answer. D cannot be the answer because D in here, the nitrile on hydrolysis is going to give you carboxylic acid. Moving ahead, let me take question number 45. Now this is based on coupling reaction. This is also based on azodie test. So here benzene diazonium salt is treated with phenol. Now as you know, over here the reaction will take place in the para position of the phenol, and what are you going to get? Basically it will remove the basically the reaction will take place over here and in the para position of phenol, and this tallies with the C option. Alright, it's a direct question based on azodie test. Moving ahead, let me take question number 46. Here methanol and ethanol can be distinguished by. Okay, so over here methanol and ethanol is given. So let's just look at the structures of methanol and ethanol. So methanol is CH3OH and ethanol is CH3 CH. Basically I can write it this way. It has this group, CH3CHOH group. So as you know, iodoform test is not only given by CH3CO group, it is also given by those alcohol which has a CH3CHOH group. Right? So this means this can be differentiated by none other than iodoform test, where the CH3CHOH part will be gradually converted to CH3COH, and then the normal iodoform reaction takes place where we get CHI3 iodoform and a CONA, right? So this can be differentiated by iodoform test. That is A. Moving ahead, let me take the 47th question here. The ether has been treated with HI, so there will be an ethereal cleavage where, what do we get? So if there is an ethereal cleavage, what do we get? So if you treat it with HI, there will be an ethereal cleavage in here. You're going to get phenol, okay, and you're going to get benzyl iodide. You're getting benzyl iodide instead of aryl iodide, because aryl iodide is not that reactive. That's why you get a benzyl iodide, okay. So over here, benzyl iodide and basically phenol are the major product. So C is the correct answer, where both A and B are obtained. Let's take the 48th question. Now this is based on the acid dehydration of this particular group. Let's see how it should be done. We have a cyclobutane ring with a CH2OH group. Let's treat it with H+. What do we get? It's an acid dehydration. We are going to convert the CH2OH in a good leaving group so that we can generate our carbocation. So basically, let's just remove the water and we got our carbocation. Now this carbocation is not that stable. There's a chance of ring expansion. So let's just take the ring expansion into consideration. So this is 1, 2, 3, 4, 5. Okay, so 1, 2, 3, 4, 5. So over here, let's just bring the plus over here. Followed by it, let the dehydration take place. And let's just put a double bond in here. So what should be the correct answer? The correct answer has to be B. The correct answer has to be A, that is cyclopentene. Alright, let me take the 49th question. Here this question is based on direct name reaction. Here as you can see over here, an aryl iodide is there and this is treated with copper. Okay, so if an aryl iodide is treated with copper, what do we get? We get this kind of a biphenyl. What is this reaction called? It is not phytic. What's phytic? Phytic will involve two aryl iodides. Okay. It will not involve aryl iodides. It will involve chloride or bromide. Ullmann reaction is the only reaction which will involve aryl iodide. Okay. So Woods Fittig and Frankland reaction is not possible. It has to be C, Ullmann reaction. Let's take the last question. Here, the electrophile attacking the ring during the reaction of phenol with chloroform and aqueous NaOH has been asked. So over here, this reaction is based on Reimer-Tiemann reaction, where if you treat chloroform with NaOH, dichlorocarbene is generated. So this matches with our B option, where dichlorocarbene is actually generated. This is called dichlorocarbene. Alright, so this is the intermediate that is used in Reimer-Tiemann reaction. Now this tallies with the B option. So these were the solutions of chemistry solutions of AIRTS Mega Test 1. I hope you have understood that. Thank you so much. Keep learning.
Hello students, let's start the zoology discussion of the paper AIRTS Mega Test 1. So to start with, let's have the first question of the section A. That is your compulsory section, right? So the first question says that Vasa efferentia leave the testis and open into dash located along dash surface of each testis. So it is from the chapter human reproduction, and we all know that our vasa efferentia leave the testis and open into epididymis. And it is located along posterior surface of each testis. So the correct answer would be your option number B. It's not urethra. So these options are absolutely incorrect. And your epididymis, it is located along posterior and not anterior. So the best option is your option B. The next question says that the regions outside the seminiferous tubules called the interstitial space contain dash cells and blood vessels. So we all know, we have studied it, that if these are seminiferous tubules that contains Sertoli cells and your germ cells here, then here in the interstitial space you have what? Interstitial cells called Leydig cells. The spelling is Leydig cells. So the correct answer for this question is option number D. Spermatid, spermatogonia and primary spermatocytes are the developmental stages of germ cells which are present within seminiferous tubules, and outside to it in the interstitial space there would be Leydig cells. So the correct answer is option D. Let's move on to the next question, question number 3, that states that in human female reproductive system, the part closer to ovaries is the funnel shaped. So just go by the shape. The funnel shaped structure in this female reproductive system is infundibulum. So the answer is infundibulum. Because the fibrae are finger-like projections. So this is incorrect. Isthmus and ampulla. Ampulla is a swollen part. This is incorrect. And isthmus is the part which is towards the uterus, right? So that would be the incorrect option. So the correct answer is option number B, infundibulum, which is a funnel-shaped structure. The next question is question 4, that says the birth canal in human females is formed by which of the following? So your birth canal comprises of your cervical canal and your vagina. So the best option is option number 1, that says vagina and cervical canal. Uterus is not a part of birth canal. So option B and C are incorrect. This is also incorrect. And oviduct, which is the fallopian tube, is also not a part of your birth canal. So the best answer and option is option A. Next question, which is question 5, is from the chapter Human Reproduction, which is about the mammary gland, that states select the option with correct sequence of structures in the mammary gland of human females. That starts with what? So if you have to start the flow of milk in the mammary gland, that have to start from the lobe, because your lobe, they are cluster of lobes. They comprises of cluster of alveoli that join to form finally tubules, and that form duct, and finally lactiferous duct, because of out of which your milk comes out, right? So you have to start your flow, you have to start your sequence from lobe. So option number B and your D becomes incorrect. Out of A and B, that starts with mammary lobe. Let's go with the flow. So from mammary lobe, it reaches into mammary alveolus. Then the milk flows to duct, then to ampulla, finally to lactiferous duct, and through nipple it comes out. So this is the correct sequence how the milk flows in this system. Third option says mammary lobe, mammary alveolus, right, but not lactiferous duct. That comes later, right? So this would be an incorrect option. So the correct answer is option A. Let's move on to the next question, which is question number 6, that says, which of the following structures is characterized by presence of antrum? So antrum is a fluid-filled structure which is well developed in the follicle, that is Graafian follicle, your mature follicle, and before that is what? Tertiary follicle. So your tertiary follicle is well characterized by the presence of this fluid-filled space, fluid-filled cavity which is antrum, right? So let's see the options first. Here you often get confused with follicle and oocyte. So please understand, you have to pay attention to these words, oocyte and follicle. And we all know that primary oocyte, secondary oocyte, tertiary oocyte. Are these the words? No, we have primary oocyte and secondary oocyte. That's it. Follicles, we have primary follicle, secondary follicle, tertiary follicle, Graafian follicle, which is your Graafian mature follicle, right? So if you talk about oocyte, your primary, secondary, tertiary follicles comprises of your primary oocyte, but the tertiary follicle, which is the matured form, that actually changes into Graafian follicle, they have secondary oocyte. And your antrum is well developed in tertiary follicle or Graafian follicle, that comprises what? Secondary oocyte. So your first option, primary oocyte, is incorrect. Tertiary follicle is the correct answer, that comprises secondary oocyte. Second is primary follicle, third is secondary oocyte. But oocyte does not have antrum. It is a follicle that have antrum, right? You have full of cells. You have this space here that comprises of oocyte within this follicle, right? So the best answer is option number B. Let's see the next question, which is question number 7, that states, read the following statements and select the correct option. So there are 4 statements given here. Let's read them carefully. The first statement says that spermatogonia, the mother cells, right? Spermatogonia, the mother cells, and secondary spermatocytes contain 23 and 46 chromosomes respectively. And this is an incorrect statement because your spermatogonia, the mother cells, they are diploid cells that have 46 chromosomes, whereas your secondary spermatocyte is a haploid structure that has 23 chromosomes. And the examiner have reversed the digits, right? So this is an incorrect statement. The next statement says formation of second polar body occurs after fertilization, which is incorrect. After fertilization, there is what? There is embryogenesis that starts. There is cleavage that starts, and not formation of second polar body. This formation of second polar body occurs when there is entry of sperm into the oocyte. So that happens before fertilization, during fertilization, not after fertilization. So this would be an incorrect statement. The third statement says the elevated circulating levels of progesterone corresponds with that of LH and FSH level, and this is also absolutely incorrect. So if you remember the levels of hormones during your menstrual cycle, you will see that during the pre-follicular phase, when the folliculogenesis is happening, you have high level of LH and FSH. Then ovulation happens where you see peak levels of your LH and FSH, and then they drop down, where actually after post-ovulation your progesterone levels starts to increase and then maintains in order to maintain the endometrium to allow the implantation of embryo. So this is incorrect because your progesterone levels do not correspond to the level of LH and FSH. The last statement says the first signal for parturition comes from fully developed fetus and placenta, which include vigorous uterine contractions. So for this statement, why it is incorrect? Because the first signal comes from fully developed fetus and placenta, that is true, but then there is hormonal regulation that induces a hypothalamus to release oxytocin from your posterior pituitary that initiates your uterine contractions in a mild level. Then a positive feedback happens that induces, this positive feedback induces more and more production of oxytocin, leading to high uterine and vigorous uterine contractions, right? So this is also incorrect. So what would be the correct answer? First and second are incorrect. No statement is incorrect. So this is wrong. 1, 2, 3, 4 statements are incorrect. This would be the correct answer. 1, 2 are incorrect. 3, 4 are correct. As no statement is correct in this question, the best answer is option number B. All statements are incorrect. Let's move on to the next question that says complete the sentence with the appropriate option, and the statement is ZIFT, which is your zygote intrafallopian tube transfer. Your zygote intrafallopian transfer. Right? So the ZIF technique, it allows the transfer of zygote or early embryo up to what stage? So as you have to transfer the zygote and embryo in fallopian tube, it has to be up to the cell of 8 blastomeres into the fallopian tube. Over and above these 8 blastomeres, the embryo have to be implanted into uterus, and that technique is your uterine transfer, right? Intrauterine transfer, where you have more than 8 blastomeres. Alright, this could be the expected question out of this. Let's see the next. So out of this, 10, 20, 16 are incorrect. C, 8 is the correct answer for this question. So the next question that says that periodic abstinence is one such method in which the couples avoid or abstain from coitus from day dash to dash of the menstrual cycle of 28 days where ovulation could be expected. This is a direct line from your NCERT, and you have to abstain from 10 to 17 days, which is the highly expected days which lie at the adjacent to your ovulation time periods in which you are highly expected to get conceived, right? So you have to abstain from such time period, and that is known as periodic abstinence. So 5 and 7, 15, 28, 2 to 10 is incorrect. The correct answer is 10 to 17. A direct line from NCERT, right? So let's move on to the next question. Question number 10 that says, which of the following contraceptive pills was developed by scientists at the CDRI, which is Central Drug Research Institute in Lucknow? The next question that could be framed from this is, the examiner may give you the pill's name and ask for it where it is developed, right? So both are important. Understand, it is CDRI in Lucknow. And the contraceptive pill was an non-steroid, it's a non-steroidal oral contraceptive pill which is known as Saheli. So the correct answer is option B. LNG20 is a hormonal IUD, Progestasert is a hormonal IUD, Multilode 375 is a copper releasing IUD. So these are incorrect. The correct answer is option B. Let's discuss the next question. Question 11 that says, which of the following is a non-medicated IUD? So we have two categories, non-medicated IUD and medicated IUD. In non-medicated IUD lies an example, lipase loop. In medicated IUD, you have hormone-releasing IUDs and copper-releasing IUDs. So the best answer for this is option number C, lipase loop, which is a non-medicated IUD. Your multi-load, copper-releasing IUD, LNG20, Progestasert, hormone-releasing IUD, they are incorrect because they are medicated IUDs. So the best answer is option C. Let's see the next question. Question 12 that says, identify the option with diseases that are not completely curable even if detected early. So we have 3H for this. Remember it this way, 3H. What these 3H are? HIV infection, herpes, and then you have hepatitis. These 3H, even if detected early, cannot be cured. Even if treated properly or detected early, they cannot be cured. So remember these 3H, it is given in your NCERT also, and it's a very important question. So these three cannot be completely cured even if detected early. So your best answer would be 1, say typhoid, tuberculosis, chlamydiasis, no. Herpes, genital warts, gonorrhea, no. Gonorrhea, syphilis, genital herpes, no. Hepatitis B, genital herpes, HIV infections. Best answer is option number D. Next question, 13. Identify the contraceptive action of copper ions in IUD. So what is the main function of these copper ions which are released in copper releasing IUD, such as your Multilode 375 and copper T and etc. So does they reduce sperm motility and fertilizing capacity of sperms? Yes, they do so. This would be the answer. Let's see other options. Decreases phagocytosis of sperms? No, they increase the phagocytosis of sperms. Distort sperm morphology? No, they don't do so. They actually initiates the inflammation immune system in this. destroys mitochondrial activity of sperm, nothing such happens. So the best answer is option 1. The next question says, Oberon of Russia and Haldane of England proposed that the first form of life could have come from. So this is from the chapter Evolution, and these two scientists, they proposed that first form of life could have come from pre-existing non-living organic molecules. And so we talked about chemical evolution. So first option, pre-existing life, non-living matter like mud and stray, that is spontaneous generation of theory. Extraterrestrials, post-panspermia, these options are incorrect. The correct answer is option C and is the answer for our question. Let's see the next question that says, question 15, S.L. Miller subjected a mixture of gases to a temperature of, and these gases, they are mixed in electric discharge sparked chamber and at a temperature of 800 degrees Celsius. So that's a direct question. Answer is option D and not the other option. This is a direct answer, right? Let's move on to the next question. Question 16 that says, another direct question, an easy question. Earth is about itne years old, and Earth is about 4,000 years old. So this is our correct answer, option C. The next question, 17, says, Ernst Haeckel proposed which of the following that supports evolution? Which of the following supports of evolution? So Ernst Haeckel have talked about the embryological evidences of evolution. So the best answer is option B, not paleontological, analogous or homologous organs. Direct question, answer is option B. Question 18 says, homologous organs are evidence of what type of evolution? So this is an example of your divergent evolution, such as your analogous organs are example of convergent evolution. Your homologous organs are example of divergent evolution. So the answer should be B, not A. Genetic different gene migration has nothing to do with it. So the best answer is option B. The next question, 19, says, which of the following moth population could not camouflage themselves in the pre-industrial period? So what happened in the pre-industrial period? In pre-industrial period, as there is no pollution, lichens were there who actually covered the trunks of trees, because of which white color have camouflaged with it. And your dark colored, dark winged, they got predated. So who doesn't get camouflaged were your dark winged one. And post industrialization, what happened? Because of the deposition of smoke and soot on the trunks, lichens couldn't able to tolerate that pollution, so they died off, and your trunk areas become darkened in color. So your dark winged moths actually get camouflaged, whereas your white winged moths got contrasted on it, because of which your predators, your birds, could actually predate on them. So post industrialization, your dark winged moths get camouflaged, whereas pre-industrialization period, your dark winged moths got predated. So the best answer here is option 1, dark winged moth, whereas white winged is, the answer should be after post industrialization. They don't get camouflaged. Checkered winged moth was not there. Pink winged moths are absolutely not there in the natural selection of such event. Let's see the next question, which is from evolution again, and a very important question. Pay attention to it. Go through the following set of animals and identify how many do not belong to, they don't belong to Australian marsupials. So you have this diagram in your NCERT that have shown your placental mammals and Australian marsupials. So let's see, we have to identify the placental ones and not the Australian marsupials. Then first is your flying squirrel. So you have your flying squirrel, which is a placental mammal. So this would be included. Then you have bobcat, again it is a placental mammal. Tasmanian tiger cat, this is your Tasmanian tiger cat, so this shouldn't be included, and it is an Australian marsupial. Then you have numbat. Numbat is an anteater, which is an Australian marsupial. Spotted cuscus is again an Australian marsupial. Flying phalanger is again a Australian marsupial. And you have to find how many do not belong to them. Answer is 2. So the best answer is 2. Right? Pay attention to it. Please remember it. This is more expected question from this chapter, right? The next question comes question 21. Human immunodeficiency virus. So the HIV, a member of the group of viruses called retroviruses, has an envelope enclosing dash genome. So it has a single-stranded RNA genome. It's a retrovirus. So the answer is RNA. A very easy question. It's not DNA, viroid and protein. So the answer is option A. Next question 22 says antigen binding site or sites on an antibody is or are found in. So you have Y-shaped 4 polypeptide chain that forms your antibody, and your variable region here, it forms the antigen binding region. This is a variable region, this is the constant region. These are your heavy chains and these are your light chains. This is a variable region of heavy and light chains. So your antigen binding site here is comprises of your variable chain of 1 heavy and 1 light chain. Right? So the best answer is both heavy and light chain. The variable region of both constitutes your antigen binding site here. Alright? So heavy chain, light chain, none of the chains, they are incorrect. It is both heavy and light chain that comprises your antigen binding site. Alright? Let's see the next question. Question 23 that says humoral response is mediated by which of the following? So humoral is like fluid. And it is actually mediated by your plasma cells. So your plasma cells, when encounters antigen, they actually differentiate into your memory T cells and then your antibodies or your plasma cells. Your plasma cells are actually effector cells. Your memory B cells were there, but they do not mediate humoral response, but they are responsible for creating this memory in your body that will allow your body to react when you have secondary immune response. Helper T cells and cytotoxic cells are part of cell-mediated immunity or CMI. So the best answer is option B. It's a very interesting question as you may get confused between option A and B. Pay attention, this response is because of plasma cells and not because of memory cells. Alright? Let's see the next question 24 that says the mode of reproduction of Plasmodium in liver cells is. So we are talking about malaria. The pathogen is Plasmodium that have 2 hosts. One is men and one is your female Anopheles mosquito. And that act as a vector also because it transmits this to your host cell, which is human, in which your asexual life actually completes. Students often get confused between asexual and sexual life. Please understand, asexual life completes in humans and sexual life completes in your mosquito. So your Plasmodium that lives in liver cells completes asexual life in human, in which it will undergo multiple fission to multiply. Fragmentation, budding and vegetative propagation are absolutely incorrect. The answer is option number C. Let's see the other question, which is question number 25 that says Haemophilus influenzae, chapter human health and disease, causes which of the following diseases in men? This Haemophilus influenzae, it causes pneumonia along with Streptococcus pneumoniae. These are the 2 bacteria that causes the disease pneumonia. It's a bacterial disease, right? It could be a viral disease also, but NCERT talked about the bacterial disease pneumonia, which is caused by Haemophilus influenzae and Streptococcus pneumoniae. Your typhoid is incorrect, caused by Salmonella typhi. Cholera is incorrect. Tuberculosis by Mycobacterium tuberculosis is incorrect. So the answer is option D. The next question 26 states that colostrum contains abundant IgA antibodies. The immunity conferred to young ones by IgA is called. So colostrum is a yellow fluid that comes out from a highly lactating mother during the initial days of lactation, and it is highly enriched in IgA antibody. So it's an example of your natural passive immunity, because preformed antibodies, IgA, they are preformed antibodies which are provided to infants for their protection, to provide immunity to them. So it's not active artificial immunity. It's not natural active because the infant is not producing it by themselves. It's not artificial passive immunity but natural passive immunity. So the answer is option D. The next question 27 says that if pairs, patches, tonsils, lymph nodes, they are what? The example of secondary lymphoid organs of a person was surgically excised and removed, what would be the physiological consequences? So what if you don't have your secondary lymphoid organs? Then there would be no place for the interaction of antibodies and antigens, right? So what would happen? Lymphocytes wouldn't mature. No, maturation happens in your primary lymphoid organs, which is your bone marrow and your thymus. Lymphocytes wouldn't be able to migrate to the site of infection. No. Lymphocytes wouldn't be able to interact with the antigen is the correct answer, because the secondary lymphoid organs are the site where your lymphocytes actually interact with the antigen. Lymphocytes would degenerate is absolutely incorrect. So the answer is option C, based on the function performed by the secondary lymphoid organs such as your pair, patches, tonsils and lymph nodes. Let's discuss the next question 28 that says Blue Revolution chapter strategies for food production. It is related to which of the following practices? Your Blue Revolution is applied for the enhancement of fish production in India. So it is for the pisciculture. Sericulture is for silkworm. Apiculture is for honeybee. Dairy farming is incorrect. The answer is for pisciculture, to enhance the production of fish in India. The next question is question 29 that says, as a consequence of MOIT, a controlled breeding technique. As a consequence of MOIT, the full form is your multi-ovulation embryo transfer technology. Its application, how many eggs are produced by the female cattle per ovulation cycle? So we know in MOIT, we super ovulate our superior breed of female by injecting FSH-like hormone into it so that it will undergo super ovulation and will produce 6 to 8 eggs in it. So the best answer is 6 to 8. So by super ovulating, this female will produce 6 to 8 eggs per ovulation cycle. 10 to 15, 50 to 60, 100, 200 are incorrect. This is the direct question and is given in your NCERT too. Excuse me. Next question 30 says, which of the following is not a part of outbreeding? Outcrossing, crossbreeding, inbreeding, mating between individuals of same breed with no common ancestors for 4 to 6 generations. Which is your outcross? Answer is option C, inbreeding, because inbreeding is not a part of outbreeding, in which mating happens between individuals of same breed with a common ancestor of 4 to 6 generations. That is your inbreeding. Next question 31 says, inbreeding depression is due to. So inbreeding depression is due to close inbreeding for many times, and this happens because of homozygosity, because it increases homozygosity. Heterozygosity is incorrect. Mutation genetic drift is nothing to apply on it. So the answer would be option 1. The next question 32 says, Jersey and Leghorn are improved varieties of. Very important questions. It's a previous year question as well, an important question from this chapter. And the answer is Jersey is an improved variety of cattle, whereas Leghorn is an improved variety of chicken or poultry. So the answer is option D. Direct question, easy question. Next question 33 says that inbreeding has which of the following advantages? So when you perform inbreeding, when you have to produce a pure line, when you have to introduce homozygosity in your line, that is where you apply inbreeding. So accumulation of superior genes and elimination of undesirable genes, no, you don't do this for this. Yes, it is a part, but just not a single part. So let's see if we have option that includes this along with some other option. Accumulation of recombinants is not associated with this. Development of pure line is also a part of this, and both A and C. So the answer is D, because it has two functions to perform: development of pure lines along with the accumulation of superior genes, and that happened by desirable selection. You will eliminate the undesirable genes. Let's see the next question. Question number 34 that says, Hisar Dale has been developed by the process of. So Hisardale is a new variety of sheep which is developed in Punjab by the cross of Bikaneri Eves and Merino rams, and it is an example of cross breeding. So example is cross breeding, answer is cross breeding. Interspecific hybridization is incorrect and its example is mule. There is no intergeneric hybridization and outcrossing is also incorrect. So the answer is option B. Let's see the next question, 35. The question says in MOIT, which is multi-ovulation embryo transfer technology, cow is administered with a hormone with FSH-like activity. So the answer is option D. Doesn't have LH, oxytocin and estrogen-like activity. Don't get confused with oxytocin, estrogen and LH. The answer is FSH-like activity. Why it is so? FSH is for follicle stimulating hormone, and what we necessitate to do in this cow is super ovulation, and to induce this ovulation, to induce multiple follicles to develop in it, we need follicle stimulating hormones, right? So the answer is option D. Let's move on to the next question, question 36. That belongs to section B. Now you have optional questions too, right? So pay attention to all the questions. The very first question says that in this section B, Apis indica, honeybee, is a species of which of the following desirable animals for animal husbandry? So it is the name of honeybee. It's not a cattle, it's not a silkworm, it's not a mule. Answer is option B. Such an easy question, right? Let's discuss the next question. Which of the following enzymes must not be used during isolation of DNA? Chapter Biotechnology, its principle and processes. So if you are isolating DNA, your genetic material is your DNA, your requirement is your DNA, then everything else is a contamination, and you will not use DNAs, because what will DNAs do? It will digest your DNA. It will fragment your DNA. So what is not required is DNAs. This is the answer. You will need RNase, protease and lysozyme. So these would be the incorrect options. Let's see the next question, 38. Identify the step that is not a part of PCR cycle. There are 3 steps in PCR. One is your degeneration that happens at 94 degrees Celsius, so that your double-stranded DNA become single-stranded DNA. The next is your primer annealing, in which the primers anneals with the respective strands, and the next is extension, polymerization, in which there would be extension of the DNA strands on the respective templates, right? So which is not a part of PCR cycle? Isolation of gene of interest. Annealing, denaturation, extension are part of PCR, so they are not the correct answer, because they are the steps which is there in the PCR. So the correct answer is option A. Let's move on to the next question, 39, that says the most common bioreactors are of dash type, and the answer is stirrer type. Fill in the blank with the appropriate option. Stagnant, non-aerated, solidified medium, they are incorrect. The answer is stirring type. The next is sparged type. There are 2 types of bioreactors which are explained in NCERT. One is sparged and one is stirrer type. And the most common type is your stirrer type. Let's see the next question. Question 40, which says which of the following genes in PBR322 is generally used as a selectable marker? So you have this PBR322 that comprises of 2 antibiotic resistance gene, which is ampicillin resistance gene and tetracycline resistance gene. So they act as selectable markers in it. In this, the options given are penicillin resistance, there is no gene for that. Chloramphenicol resistance, kanamycin resistance, there are no genes for that. The answer is ampicillin resistance. The other option could be tetracycline resistance also, that would be the correct answer also. So the answer is option B. Please understand, this plasmid is really important. The sites given in this plasmid are also equally important. We'll see some questions like that. The next question says, identify the type of ion used to make the host competent to enable it to take up the plasmid. So before transformation, we have to form this host. We have to prepare this host as a competent cell. Competent cells are those which are prepared to take up this DNA. So in order to make them competent, what we need to do is we need to treat them with divalent cations, right? Divalent cations such as calcium ions. So your monovalent cation is incorrect, monovalent ion is incorrect, trivalent cation incorrect. Answer is divalent cation. The example is calcium. So this is the most asked question. If they will ask from this topic, the cation is your calcium ions. The next question says, in PCR polymerase chain reaction, the DNA polymerase employed for the extension of the strands is called. So for PCR, as it starts with the step of denaturation that happens at 94 degrees Celsius, you need a DNA polymerase that won't get denatured at such a high temperature. And that polymerase is your Taq polymerase. Why Taq? Because it is extracted out from your thermus aquaticus. So T for thermus, aquaticus, AQ from this. So it's a Taq polymerase. It's a thermophilic enzyme that can sustain at such a high temperature. So we use Taq polymerase. Reverse transcriptase is an enzyme which is there in HIV virus. DNA ligase are used to ligate different strands of DNA. DNA polymerase 3 is another DNA polymerase enzyme that are used in cells. So for this PCR we use Taq polymerase. So the answer is option B. Let's see the next question, 43, that says micro-injection method is most suitable for. It's a moderate kind of question because it is there, but you have to find out a very close answer from these options. Micro-injection method is most suitable for. Is it suitable for animal cell? Yes, because in micro-injection you will directly inject your gene of interest into the cell. Plant cell, no, there is cell wall in it. Yeast cell also has cell wall in it and not bacterial cell. So the answer is option A. Let's see the next question, question 44, that says transgenic animals have been created to produce useful biological products like alpha-1 antitrypsin that is used for the treatment of. And this alpha-1 antitrypsin is used for the treatment of emphysema. So please relate it to your class 11 chapter breathing and exchange of gases, where you have seen this disease or disorder of respiratory passage, which is emphysema, in which your alveolar walls get damaged. So the answer is emphysema. Typhoid, pneumonia, and tuberculosis are incorrect options, and the correct answer is option B. Let's discuss the next question, 45. In autoradiography, which of the following statements is correct for the clone having the mutated gene? Please understand, it is again a very confused thing. Students often get confused that if it's a mutated gene, and if we are looking at it in autoradiography, we should look for the mutated gene. But opposite happens. When you look at the film of your autoradiography, you will not see mutated gene at all. And that is how you understand that at this place, as there is nothing, this is your mutated gene. So for this mutated gene, it will not appear on your photographic film is the correct answer. Very confusing thing. Please understand it and take a note. It will appear on the photographic film, no. May or may not, no. It will never appear in it. It will get degraded or damaged is incorrect. So the correct answer is option B. Let's see the next question. Question 46. The first clinical trial for gene therapy was done for which of the following enzyme deficiencies? So it is done in a 4-year-old girl and that is done against a deficiency of adenosine deaminase. And finally that lead to SCID. So the answer is deaminase, not transaminase, kinase and phosphatase. They are incorrect. The answer is deaminase, option number C. Let's move on to the next question, which is question number 47, that says identify the correct statement for the production of recombinant human insulin or humulin. The first statement says A and B peptides were produced together. No, they are produced separately. C peptide was intact in active humulin. No, there is no C peptide in the mature form of insulin humulin. A and B peptides were produced separately is the correct answer. Presence of C peptide made no difference to the activation of humulin is also incorrect. Right? So we have A and B chains. that are produced separately, and this will form your hemulin. There is no C polypeptide chain in this mature form of hemulin. Excuse me. Next question, question 48 says, at present, how many recombinant therapeutics have been approved for human use the world over? So understand, it is different. The digit is different from if you talk about world and if you talk about India. In together, there are 30 recombinant therapeutics that are used worldwide, but in India it is just 12. So if the question is asking for worldwide, the answer would be D, not 10, 20 or 40. And it is 12 if you talk about India. Alright, remember this too. Let's see the next question, question 45, that says, in India, how many recombinant therapeutics are being presently marketed? So we have just discussed it, and the answer is 12 directly, not 11, 10 and 9. So the answer is option number A. Let's see the last question of the subject and this category, this section, which is question number 50, that says, the process of RNA interference, RNAi, is used by creating double-stranded RNA for gene silencing, is used to prevent infestation of. So we all know this, that it is to prevent the infestation of a nematode, an ASCII helminthes, which is Melidogyne incognita. Agrobacterium tumefaciens is a bacteria. This is incorrect. Melidogyne incognita is the correct answer. E. coli is a bacteria. Bacillus thuringiensis is a bacteria. So these are incorrect options. The correct answer is option number B. So I hope you understood it and be thorough with this. These concepts are really critical and are about to come in your maybe next NEET. So all the best for the next exam.
Hello students, how are you all? Today we are going to discuss about this AIRTS Mega Test 1, and here we are going to discuss about botany section. As you all know, these AIRTS of NEET has started on 28th of November, and today we are going to discuss about Mega Test 1 here. This is the schedule here. We are talking here about the Mega Test 1, and in this session we are going to talk about botany section. So let's get started. First question here, students, is choose odd one out with respect to ploidy of structures in the flowering plants. So here we have to discuss about the ploidy. And we have to select the option which will be odd one out here. So if we talk about the first option, ovary, second is anther, C is zygote, and D is pollen grain. So some of them are having same ploidy here, and one will be having the odd ploidy here. If we talk about the ovary, it is having here diploid. It is 2n in nature. And we talk about the anther, it is also 2n, diploid. Zygote here is diploid, 2n. Whereas if we talk about the pollen grains, they are haploid. So obviously, odd one out here is pollen grains. D option is correct here. Second question, majority of sexually reproducing organisms, they produce. What we are talking about? Sexually reproducing organism. And majority of them, what kind of gametes they are producing? First one, we are having homogametes. Obviously, morphologically, in the form and structure, they will be similar. Isogametes, they are also similar kinds of gametes. C is heterogametes. Heterogametes, obviously, they are different in form and structure. I can write here different form and structure. So majority of the gametes produced by the sexually reproducing organism, if we can show here, the small one and the larger one. Larger one, many of the time it is immotile, and the smaller one, it will be mobile here. So these are kind of heterogametes we are having. So majority of them, they are producing C, heterogametes. And if we talk about the D option, it is saying that morphologically indistinct gametes. Obviously, this is the incorrect option. Why? Because it is saying that indistinct. Whereas majority of the sexually reproducing organisms, they are producing heterogametes, or we can say morphologically distinct gametes. We can say that they are distinct here, not indistinct. So our option C is correct here. Another question, select the correct statement with respect to structures of microsporangium. Here we are talking about microsporangium, and we have to see the correct statement. First one is saying A generally surrounded by 3 wall layers. If we see the image here, see here, this is the section of microsporangium in case of flowering plants. So we are having here the 4 layers. It is surrounded by the 4 layers, not 3 layers, 3 wall layers. So obviously this option is incorrect. What are the layers here? Epidermis, endothecium, teptum, and middle layers. So 4 wall layers are present in this microsporangium. B is saying inner 3 wall layers perform the function of protection. So obviously this is also the incorrect because nutritive layer is also present there. C is saying teptum is the outermost wall layer. Is it? Teptum is here. So obviously it is not the outermost. We are having also the epidermis, endothecium here. So obviously this option is incorrect. If we talk about the teptal cells, as you can see here, teptum, it is having its cells. Now we talk about the teptal cells, they possess dense cytoplasm, it is correct here, and generally have more than one nucleus. So they are having nuclei. Number of nucleus will be present here. Obviously correct option here is D. A, B, C, they are incorrect. Another question here, select the mismatched pair. A is sporopollenin, that is exine. This is correct match here. We have to see the mismatched. B is saying that bronchial afflictions with respect to the pollens of entomophilous flowers or entomophilous plants. What are they? They are insect pollinated flowers. They are insect pollinated flowers here. And if we talk about the bronchial affliction, if we talk about the bronchial afflictions, what are they? This is the inflammation of the bronchi. So inflammation of the bronchi is associated with anemophily. Anemophily means the air pollinated, that the pollination will be occurring through wind or air. So obviously bronchial affliction or the inflammation of the bronchi is related to the anemophily, not here the entomophilous flowers or entomophilous kind of plants. C is saying nutritive wall layer. Tapetum, as we have seen here also, the tapetum, it was nutrition rich layer. This is also correctly matched. D is basal part of ovule is chalaza here. As you can see here, this is the figure of our ovule and the basal part here. What it is representing? It is representing here chalaza. So obviously A, C and D, they are correctly matched here, whereas the B one, that is inflammation of the bronchi, will be related to the anemophily or the wind pollination. Another question we are having here in transcription in eukaryotes. We are talking here about the eukaryotic transcription. The RNA polymerase 1, it transcribes, we are having here the options. It will transcribe precursor of mRNA or hnRNA. B saying mRNA with additional processing like capping and tailing. C saying tRNA, 5S ribosomal RNA and snRNA. Whereas D saying 28S, 18S and 5-S ribosomal RNA. So obviously this RNA polymerase 1 is related to the transcription of the ribosomal RNA here. So D option here is correct. Another question, statement A, primary transcript contain both exons and introns, in which introns are the non-coding part. Here we are talking about the primary transcript. Now it is having exons as well as the introns. Remember students, when we are talking about the exons, they are the expressible parts. Means that we will have the formation of protein out of them. So exons and introns. And introns, they are in between the exons and they are non-coding part. So obviously in the primary transcript, we are having the presence of introns as well as the exons here. Statement B is saying process of capping and tailing. As you can see here, this is capping and tailing. It represents the dominance of RNA world. So here we can say that capping and tailing, it is related to the action of the nucleus. We have to prevent this newly transcribed RNA from the nucleases. That is why we are having this processes such as tailing and capping. So statement B here, it will be incorrect. We are having only A as correct. That is, exons and introns will be present in the primary transcript. Another question, which of the following characters cannot be studied in an individual? Here we are talking about an individual here. Now we are having sickle cell anemia. Some of the genetic disorders, they have been given here. First is sickle cell anemia. Second is phenylketonuria. C, ABO blood groups and thalassemia. So when we have to study these genetic disorders, can we study sickle cell anemia and we can identify in an individual? Obviously we can. If we talk about the phenylketonuria, this also we can study in an individual. If we talk about the C, ABO blood groups. So obviously this is related to multiple allelism. So it cannot be studied, these characters cannot be studied in an individual because an individual will be having either A blood group or B or AB or O. So an individual, only a person is not having all the blood groups. So obviously C is the correct answer here. That is the ABO blood groups. They cannot be studied only in an individual. C is here our correct option. Another question, select the incorrect statement. We have to see the incorrect statement. A saying human male is heterogametic. As you can see here, we are having the humans. And in the females, we are having these two X chromosomes. If we talk about the male, we are having XY. Obviously, this is also the case for Drosophila. So heterogamety we are seeing in the human male. You can see here XY is present in the human male. This is correct here. In male grasshopper, 50% of sperms have no sex chromosome here. 50%. So if we talk about the male grasshopper, see here. This is also the correct here option. We are having here XO. So 50% of the sperms, because we are having this combination. And when we are having the gametes, they will be like this. So this statement is also correct here. Another statement we are having, human males have one of their sex chromosome much shorter than the other. Here we are talking about the Y chromosome. Y we are talking about. This is also correct here. And if we talk about the last statement here, female birds are homogametic and have ZZ chromosome. If we talk about the bird in the females, birds and female we are having. Is it showing homogamety or heterogamety? Obviously, it is showing here ZW, that is the heterogamety, as we have seen in the case of human male. So we are having this option correct, as this is the incorrect statement here. Female birds, they are heterogametic and they are having ZW kind of chromosomes. Another question, biogas production technology in India was developed mainly due to the efforts of. We are having the options as KVIC and NSC, ICAR and KVIC, IRRI, and D is IARI and KVIC. So obviously this D option here is correct. This is the Indian Agricultural Research Institute and Khadi and Village Industrial Corporation we are having, that is KVIC. Because of that, biogas production has been introduced in India, this technology, where we are having the production of the methane. Mainly the biogas is having the methane, and because of the digestion by the methanogens, which will be producing here CH4. So correct option is D1 here. Another question, in mung bean, resistance to yellow mosaic virus, what kind of diseases and powdery mildew were brought about by? So certain varieties they have been produced. In case this here we are having the mung bean, and they have developed resistance against certain diseases such as yellow mosaic virus and powdery mildew. And this is because of the technology we are calling as mutation breeding. So we are having options as hybridization and selection. C is tissue culture, biofortification. Remember students, when we were talking about the biofortification, we have developed some varieties that are rich in vitamins and minerals. So this is not the case here. We are having mutation breeding. Because of that, certain varieties, they have developed resistance against certain diseases here. So mutation breeding, what we are doing in this? We are having the application of radiations as well as the chemicals. And they are giving here mutation. They are inducing here mutations. And these some varieties which will be having these mutations because of these radiations, chemicals, they will be acquiring resistance against these diseases. So mutation breeding, by this we have developed these varieties against disease resistance. So correct option here is B. Another question, statement A saying all organisms show behavioral responses to cope up with variations in their environment. So we are here talking about all organism. Is it correct? Obviously not. Some organisms, they are having these responses, behavioral responses. Because of that, they are able to cope up with different kinds of environments. But can we say about all organisms? No. So statement A here is incorrect. Another statement is archaebacteria flourish in hot springs and deep sea hydrothermal vents where temperature is very low. So you have to read the statements very carefully. Obviously, here in these conditions, we are talking here about hot springs and deep sea hydrothermal vents. So obviously here the temperatures, they will be quite high, not very low. So both the statements, they are incorrect here. So we are having the correct option as D. Both statement A and B are incorrect. Another question, marginal leaf buds are present in. We are having the options as potato, ginger, agave and bryophyllum. So we are here talking about the marginal leaf buds. Leaf buds, they will be present on the margins. As we can see here, this is the example of bryophyllum. So we have studied it in the vegetative reproduction. Helping in vegetative reproduction. As in the case of these, you can see here, these are the buds which will again give rise to new plant. And we can say that this is vegetative reproduction as we have seen in the case of bryophyllum. Another question, a high BOD in sewage water, it indicates. So if we talk about the BOD, as you can see here in this graph, we are having this BOD. What do we mean by that? Obviously it is biological, oxygen demand. So when BOD is high, we mean that water body is quite polluted, and because of that we are having here the microorganisms, they will be requiring a lot of oxygen to degrade that pollutants, or we can say organic matter present in that water body. So if BOD is high, means that water body is quite polluted here. So we are having options, high content of inorganic material. When we are talking about the BOD, we are talking here about the oxygen demand by the microorganisms, and they are acting on the organic material, not on the inorganic material. So our first option is incorrect here. We are not here talking about the inorganic material. B is saying more polluting potential. So obviously students, when BOD is high, we mean that organic material or the pollutants, they are quite high in that water body. And that is why the microorganisms, they are requiring a lot of oxygen to degrade that organic material. So we can say that water body is having more polluting potential. It is having a lot of pollutants in we can say that in presence of organic material. So B option here is correct. If we see C and D, low content of organic material, that is wrong. Why? High BOD, it is representing here high organic material on which these microorganisms will act upon. D is high dissolved oxygen. This is having inverse relationship with this BOD or the biological oxygen demand here. So DO, dissolved oxygen, and BOD, they are inversely related here. So we can say that high BOD, it is representing here low dissolved oxygen. So our option B is correct here. Another question. First is biogas is a mixture of gases containing predominantly methane. If we talk about the biogas, obviously it is a mixture, but we are having high proportions of methane, or we can say that CH4. So this statement is correct here. Another statement is saying that methanogens are methane producing eukaryotic organisms. When we are talking about the biogas production, what we are having there? We are having there methanogens. And obviously they are the methane producing organisms, but they are not eukaryotic. They are prokaryotic organisms. When we were talking about the Monera, what we have seen there, we were having the bacteria, archaea, and these methanogens there also. So obviously these are the prokaryotic organisms that will be producing methane here in the biogas production. 3 is saying methanogens are obligate or strict anaerobic bacteria. Obviously they are bacteria, they are anaerobic, they are working in the absence of oxygen, and they are obligate or strictly anaerobic kind of bacteria. So this statement here is also correct. So we have to find here how many of the above statements they are true for biogas production. So we are having first and third, they are correct. Only second one is incorrect. So we are having C option is correct, where 1 and third, they are the correct statements. Another question, cyanobacteria are used as biofertilizers as they. Cyanobacteria, we know that blue-green algae. They are also used as the biofertilizers. That is, for the growth of plants, they are being used. And because they are organisms, living organisms, we are calling them as biofertilizers. So what they are helping the plants with? We are having the correct option. We are having here the options as can fix atmospheric nitrogen, have microscopic structure that is not useful for the plants, so we cannot say that, are aquatic organisms, are autotrophic organisms. So obviously when they are being utilized as biofertilizers, they are giving something to the plant. So they are fixing the atmospheric nitrogen in forms of nitrogen that can be used by the plants. So certain forms they are producing, they are converting the atmospheric nitrogen in these forms which can be further utilized by the plants. That is how we can say that they are fixing the nitrogen, and that is why they are known as biofertilizers, these blue-green algae or the cyanobacteria. So we are having the correct option here as A. Another question, which of the following is the commercial blood cholesterol lowering agent here? So we are having the utilization of some microorganisms, and we are having production of this blood cholesterol lowering agent which is known as statins. So we are having streptokinase, cyclosporin A, statin and proteases. So microorganisms, they have been utilized and they are producing these blood cholesterol lowering agent which is also known as statin here. So C option here is correct. Another question, statement A. Trichoderma is a bacteria used as a biocontrol agent in the treatment of plant diseases. So trichoderma is a bacteria, it is saying that, and it is used as biocontrol agent in the treatment of plant diseases. So obviously this is not the correct option here. This statement is incorrect. And we are having conversion of milk to curd improves its nutritional value as it increases the amount of calcium. Is it so? Obviously not, students, when we are having the conversion of this milk to curd. And we can say that nutritional value is increasing. But how? Is it increasing the calcium level? Obviously not. Vitamin, which is B12. This is increasing in the curd, and because of that, nutritional value of curd is more than the milk. So this statement is also incorrect here, and the first one is also incorrect here. Why? Trichoderma is basically a fungus. So both the statements, they are incorrect here. Correct option will be both statements A and B, they are incorrect. Another question, skin color in humans is an example of. So we are having certain allele interactions here and gene interactions here. They are codominance, incomplete dominance, pleiotropy, and polygenic inheritance. When we talk about the skin color in humans, you can see here we are having this representation. So as you can see here, students, as you can see here, we are having combination of different genes. And because of that, we are having a range or grading of skin color. You can see here the darker color, darker, and it is going lighter here. So grading of color in this case is happening. And it is dependent on different kinds of genes. So different genes, they are interacting with each other and they are responsible for one character here. One character, but there is grading, or we can say that different intensities of that character is there. So different genes, they are interacting together and they are contributing to these intensities of this one kind of character. So this is the example of here polygenic inheritance. So skin color in case of humans, as you can see here in this cross, this is the example of polygenic. As I've told you, different genes, they are interacting and contributing to one character. So we can say that this is the example of polygenic inheritance. That is why this D option here is correct. Another question, in which of the following Mendelian disorder enzyme required for phenylalanine to tyrosine conversion? What is not happening in this kind of disease? Phenylalanine is not being converted to tyrosine. This is stopped in this kind of disease. We have to find out which kind of disease it is happening. This is the characteristic of what? We are having here hemophilia, thalassemia, phenylketonuria, and cystic fibrosis. So by the term itself, we are clear here, this must be phenylketonuria. What is happening here? Accumulation of phenylalanine. It happens. As we have already studied that phenylalanine is not being converted to this tyrosine, and because of that accumulation of this phenylalanine will happen, and this is the reason we are having this disease phenylketonuria here. This is also a genetic disorder. So correct option here is C. Another question, choose odd one out with respect to limitations of ecological pyramid. Here you can see we are having this ecological pyramid and we are having certain limitations to that. But which one is not correct with respect to these limitations of the ecological pyramid? We are having here the options. Does not consider the same species belonging to two or more trophic levels. This is correct for ecological pyramid, as we are having the different levels. You can see here trophic levels, producer, herbivore, carnivore, and the top carnivore here. So when we are making an ecological pyramid, it is not considering the same species which will be belonging to different trophic levels. As we know that in the food web, we can have single species. It can be at one trophic level as well as it can be at another trophic level. But here in the ecological pyramid, we are not considering that. Another option here considers simple food chain. So obviously, we are having 1, 2, 3, 4 trophic levels as we have seen in the case of food chain. So we can say that food web is not being represented here in this ecological pyramid. C is saying saprophytes are given space. This is not correct here. Saprophytes, they are also not given space in this ecological pyramid. D is does not accommodate food web. As we were talking about these two options also. So this is the same here. Food web is not considered. It is based on the simple food chain. So C option here is correct. This is the odd one out here. Another question, The entire sequence of communities that successively change in an area is known as, here we are talking about the ecological succession. So entire sequence of the communities that will successively change in an area. As you can see here, this is the barren rocks, lichens, mosses. Obviously these will be here are pioneer species. And this will be the climax community, the last one. But the sequence which will be changing, the sequence of the communities here, they are known as seral stages here. So we are having the climax community, it will be at the end. Sear, obviously this is the correct option here. Communities that will be keep on changing. Pioneer community, this is the first one after the barren rocks, and D is transition community. We are not having this term in this ecological succession, so B is the correct option here. Another question, in which of the following population interaction parasitic bird lays its egg in the nest of its host and let the host incubate them? So here we are having one parasitic bird. This is the example of one kind of interaction we are having, and it will be laying the eggs in the nest of the host bird. So we are having this example in case of cuckoo and crow. So cuckoo lays its egg in the nest of the crow and crow will incubate them. So this is a kind of interaction which is known as parasitism. And parasitism is what kind of interaction? Plus, minus. So obviously cuckoo will be benefiting here, whereas the crow, it will be expanding its resources here. So it will be having some harm on itself. So we are having this parasitism and we call it as brood parasitism. So we are having correct option here is brood parasitism, plus minus kind of interaction here. Another question. First statement here is major constituent of biological membranes and nucleic acids. Here we are talking about one, we can say that element here. So we have to find out on the basis of these statements which one is correct. Major constituent of biological membranes and nucleic acids. So if we talk about the nucleic acids. Second is saying natural reservoir is rock here. So is the carbon having natural reservoir as rock? Obviously not. We can eliminate it. Hydrogen, it is not having the natural reservoir as rock. And if we talk about the oxygen, obviously it is also not having natural reservoir as rock. So we are having here the phosphorus only. But if we see the third statements, herbivore and other animals obtain it from plants. So phosphorus is the element we are having which is already present in the nucleic acids, that is DNA, RNA, and in the membranes, cellular membranes also. And it is present in the rocks, natural reservoir we are having, and in the case of phosphates as phosphate. And third one also, the animals, they are obtaining it from the plants. So we are having the correct option as C. Another question, CNG is better than diesel and petrol because it A, burns less efficiently unlike petrol and diesel. When it is burning less efficiently, why it will be very useful, or we can say better than the petrol and diesel. So obviously this statement is incorrect. Is costlier than diesel and petrol. This is not the correct option here. Cannot be adulterated. It is CNG is better because it cannot be adulterated like petrol and diesel. C option here is correct, all except C. So this one is also incorrect. We are having the correct option as C. Another question, absence of one sex chromosome here, it causes Down syndrome, Klinefelter syndrome, Turner syndrome, and Cry-Do-Cat syndrome. Here we are talking about one of the genetic disorder, and in that what condition we are having? absence of one sex chromosome. So we are having this in the case of Turner. If we see the karyotype here, we are having the autosomes here and sex chromosome. We are having here only one. XO condition is here. So obviously this is the case for Turner syndrome, and based on that we are having certain characteristics. Another question, how many of the given Mendelian disorders can express equally in male and females? When any genetic disorder will be expressed equally in male and female? When it will not be associated with X or Y chromosome. So we have to see here expressed equally in male and females. If we talk about the hemophilia, color blindness, thalassemia, phenylketonuria and sickle cell anemia, out of them, which one are related to the X or Y chromosome? Obviously students, hemophilia and color blindness, they are X related here. X-related genetic disorders, that is why they will not be expressed equally in male and female. Obviously, thalassemia, phenylketonuria and sickle cell anemia, they will be expressed equally because they are not related to X or Y sex chromosomes here. So they will be equally expressed in the male and females. So we are having 3 out of these 5, they are expressed equally here. Another question, first statements, technique developed by Alex Jeffrey. When we are talking about a technique which is developed by Alex Jeffrey, we are here talking about the DNA fingerprinting. Second statements, radioactive DNA segments are used as probe. So we are using the probes and they are radioactive DNA segments. This is also correct here. It is for the detection of DNA regions showing very less degree of polymorphism. Polymorphism, when we are having different kinds of bases, nucleotides here, we are having this polymorphism. So we are having this DNA fingerprinting and it is based on the right statement here will be it is for the detection of DNA regions showing high degree of polymorphism here. So this statement is correct here, incorrect here. Fourth is southern blot hybridization step is used. This is also correct here. So how many of the above characters are true for DNA fingerprinting? So obviously here first, second and fourth. 3 they are correct here. We have to see true for DNA fingerprinting. So 3 they are correct here. And third one is incorrect because it is saying that it is for the detection of DNA regions which are showing very less. But instead of that, DNA fingerprinting is based on high degree of polymorphism. Another question, choose the correct option with respect to emencelism here. Species A and species B. We have to see emencelism is what kind of interaction. Plus and minus. We have seen that in the case of parasitism, minus and 0 and plus and minus. That we will see in the case of competition. So we have B and C options. When we talk about the emencelism, this is second kind of interaction. One species here will be harmed and another species will not have any effect. If I talk about the example of this emencelism, we can say that interaction between the penicillium, which is a fungi, and it will be producing here penicillin. And this penicillin will be acting on the bacteria. And will be harmful for the bacteria, whereas penicillium will not be having any effect over it. So this minus 0 kind of interaction is emencelism as given by this example. So B option here is correct. Another question, we are having the statements here. Statement A, greenhouse gases absorb long wave radiations reflected from the earth and then emitted again towards the earth. So we are having here this phenomena of greenhouse effect. We are having here, see here, long wave radiations, they are being reflected back. As this is the statement, greenhouse gases absorb long wave radiations reflected from the earth and then emit it again towards the earth. That is why we are having increase in the temperature. That is why the temperature of the earth is habitable for the living organisms, because of this greenhouse effect. Another statement, greenhouse effect is artificially occurring phenomenon that is responsible for heating of Earth's surface and atmosphere. This statement here is correct, heating of Earth's surface and the atmosphere, but it is not the artificially occurring phenomenon. We can say that this greenhouse effect, it is a naturally occurring phenomenon, not an artificial one. So we can say that this statement is incorrect here. Only the statement A is correct. So we are having the correct option as only statement A is correct. Another question, the second trophic level in the lake usually comprises of, we are here talking about the ecological pyramid or the food chain. So we have to see the second trophic level. A is benthos, B fishes, zooplanktons, and D seagull. When we are talking about the ecological pyramid of a lake, at the first trophic level, obviously we will be having the producers, then the consumers, and top consumers, then we will be having. So producers, they are phytoplanktons. They are the planktonic species, but they are producers here. Above them, they are zooplanktons. Planktonic species, but they are dependent on these producers. They are basically the consumers here. So we are having the second trophic level of the zooplanktons. Correct option here will be C. Another question, in the cell of fungi and plants, RNA splicing, capping and tailing. As we have seen earlier also, the case of capping and tailing. And when we talk about the splicing, what we are having, the removal of the introns. Introns, non-coding sequences, they will be removed. We are having ultimately the processing of the RNA here. So the introns will be removed through splicing and capping and tailing to prevent from the nucleases, the RNA. Newly transcribed RNA we are having. It takes place inside. We are talking here about the eukaryotes. Cytoplasm, nucleus, nucleolus and dictyosomes. Obviously, we are having this processing of the RNA. As we can see here, we are having this addition of phosphates here and the tailing here and the capping here in the nucleus. In case of eukaryotes, we are having fungi and plants, as this example is given here. So correct option here is B. Another question, identify the inheritance pattern in the given pedigree. So we have been given here the pedigree. See here students, we talk about the options. A is Y-linked. If we look at this pedigree, if the straight, we can say that the colored blocks or the black ones, they are infected individuals here. If this disease was Y-linked here, this would have been the case here. Obviously not. This is for the female and this is for the male here. So this option is eliminated from it. We are not having this disease as Y-linked. B, autosomal recessive, C, autosomal dominant, and D is autosomal recessive. We can say that these two options, they are similar. So obviously this will not be the option here. And if we have to solve this pedigree, see here, we are taking it as autosomal dominant here. I can say that this will be the case. In this, if it would have been capital A and capital A, small a, all the offsprings, they would have been infected here, or they would have been diseased here. But this is not the case here, because this one is unaffected here. So we will have here this combination, and I can say that this is the uncolored one is representing here this condition, because it is not affected. So if we talk about this, again it is being crossed with unaffected one, we are having this combination. By the options we can understand this is the example of autosomal dominant here. That is why the offsprings, they are having these combinations. So C option here is correct. Another question here is, we are having 4 statements. We have to see which one is correct and which one is not. Is hemophilia is an autosomal recessive disease? As we were talking earlier, hemophilia was X-linked recessive trait here. So this is the incorrect one. Down syndrome is due to trisomy of autosome. Trisomy of 21st chromosome we are having. This is the correct statement here. Third is saying phenylketonuria is an autosomal recessive gene disorder. So we are having phenylketonuria, which is autosomal recessive gene disorder. We will see if it is correct or not. D is sickle cell anemia is an X-linked recessive gene disorder. So we are having the incorrect 2 statements here, and the 2 are correct. Which one are correct? Second and third, they are correct here. Phenyl is autosomal recessive genetic disorder, whereas Down syndrome is a trisomy of 21st chromosome. Whereas in the case of hemophilia is X-linked recessive disorder, and sickle cell anemia is autosomal recessive disorder here. So they have been exchanged here, first and fourth. How many of the above statements they are correct? Only they are second and third. So C option here is correct. Another question, we are having the 2 statements. Statement A is saying desert lizard lacks physiological ability to deal with high temperature of their habitat. Are they lacking? Obviously not. They are having here the we are having the statement here, and we are talking here about the physiological ability here. Whereas we have seen in the case of adaptations, what kind of adaptations they are having? Desert lizards. They are having adaptations related to their behavior. I can write it here. Behavioral adaptations they have seen to cope up with the extreme environments, as in the case of high temperature. They hibernate, we can say that, to undergo very extreme temperatures, to hide from the extreme temperatures. And the statement B is saying, mammals from colder climate generally have shorter ears and limbs to maximize heat loss. Is it correct here? As we have seen in the case of rabbits, which were present in the polar areas or the Arctic areas, and which were present in the tropical areas. So we have to compare mammals, as we have seen in the example of rabbit, on the polar and the equatorial or the tropical areas. These were having shorter ears and limbs. Is it to maximize heat loss? Obviously not. This is to minimize the heat loss. So this statement here, B, is incorrect. We are having the correct statement only as statement A, which is saying that they are lacking the physiological adaptations, because they are having certain behavioral adaptations that they will be requiring to cope with the maximum temperature or the high temperatures where they are found. So A statement here is correct, and the A option here is correct. Another question, which of the following are the important steps in the process of decomposition? We are having the decomposition process, which is being done by the microorganisms. We are having certain combinations here. As you can see here, the steps in the decomposition. What are they? They are the fragmentation, leaching, catabolism, humidification, and mineralization here. Is detritus fragmentation only? Obviously not. We are saying that only. Fragmentation, catabolism and leaching. Productivity and humidification only. And energy flow and mineralization. Obviously this will be the correct combination here, as we are having the fragmentation, leaching and catabolism, as well as the humidification and mineralization. So we are having the most appropriate here, the B option. So the correct here is B. Another question, mark the incorrect statement. A is exons are defined as coding sequences. So when we are talking about the exons, expressible strengths, you can remember from it, that will be converted or that will be forming the proteins ultimately, or the amino acids ultimately. B is DNA segment that code for ribosomal RNA, and tRNA here are also considered as gene. This is also the correct here. We have to see the incorrect one. RNA polymerase 2 transcribes precursor of mRNA. When we were talking about the one, it was transcribing their ribosomal RNA. Whereas the RNA polymerase 2 is going for the mRNA or the messenger RNA. D is transcription, translation and replication can be coupled in bacteria. When we talk about the coupling, because the bacteria, they are prokaryotes. So processes such as transcription, translation, not the replication, they are coupled in these prokaryotic or the simple organisms here. So correct option here is D. Another question, choose odd one out with respect to conservation strategy. As we know that we are having two modes of conservation, and what are they? They are in situ and ex situ. When the organisms, they are being protected at their own place and protecting the outside ecosystem or the outside environment also, we are calling this kind of conservation strategy as in situ. Whereas if they are being protected in their artificial environment, we are saying it as ex situ kind of conservation strategy. So we are having the options zoological park, botanical garden, pollen bank and national park. We are talking about the zoological, botanical and pollen bank. They are all related to ex situ. Artificial kind of conservation strategy we are having, whereas the national parks, they are in situ. So odd one out will be here, national parks. Another question, which of the following step is crucial for the success of plant breeding experiment? When we are doing the plant breeding, we are having certain steps. First, we have to see the crucial one. A, evaluation and selection of parents. B, collection of variability. C, selection and testing of superior recombinants. And D, cross hybridization among selected plants. So we are having the selection and testing of the superior recombinants, as we are having certain characters that will be valuable for us, as you have seen in the case of disease resistance, drought resistance. So obviously when we are having these combination, so we have to do this testing and the selection of the superior recombinants. This is the crucial step for this plant breeding experiment, and this would ultimately lead to a development of better quality of the variety of the plant. So we can say that C option here is correct. Another question, which of the following statement is incorrect for organism used as biocontrol agent? A is ladybird is used to get rid of aphids and mosquitoes. As you can see here in the image also, we are having this ladybird and it is feeding on these aphids. So this is the correct statement here, and it is a mechanism of biocontrol, or they are also working as biocontrol agents. So this option or this statement here is correct. Some of the baculoviruses, which are major plant pathogens, belong to genus Nucleopolyhedrovirus. Remember students, when we are talking about the baculoviruses, obviously they are related to these Nucleopolyhedrovirus. but they are not the plant pathogens here. They are feeding on the insects. They are feeding on the insects or aphids, we can say. So this statement here is incorrect. Microbial biocontrol agents can be introduced to control butterfly caterpillar here. So obviously, this is also the correct statement here. A way of biocontrolling here. They are working as the biocontrol agents. Trichoderma is effective against several plant pathogens. One of the example of this biocontrol. So this is option here is also correct. We are having the incorrect option as B, because baculovirus, it is attacking on the insects. It is feeding on the insects, not it is a plant pathogen. That is why it has been utilized as a biocontrol agent. Another question, good ozone is present in A, upper part of atmosphere, B, lower part of atmosphere, C, ionosphere, and all except A. When we are talking about the good ozone, see here, as you can see in this image, good ozone and the bad ozone. When we are talking about the good ozone, it is protecting our environment, our living forms from these harmful rays, UV radiations. That is why it has been known as good ozone. Where it is present, we are having this earth and we are having these layers, stratosphere, thermosphere, ionosphere we are having. We know that this ozone layer is present in the stratosphere. So it is near to the upper part of the atmosphere. So obviously, good ozone, it is present in the upper part of the atmosphere. A option here is correct. If we talk about the bad ozone, it is affecting the health of the living organisms here present on the earth. That is why it has been termed as bad ozone, and it is nearby the earth's surface, lower part of the atmosphere. So A option here is correct. Which of the following type of pollination is rare in flowering plant and is limited to 30 genera? Mostly monocots. We are having anemophily, that is wind pollination, hydrophily, water pollinated, entomophily, that is related to the insects, and ophiophily, that is related to the snails here. So we are having this hydrophily, which is the water pollinated, or the monocots which will be pollinated with the help of water. That is quite limited here, is rare in flowering plants. So we are having the correct option as B. As you can see here in Vallisneria, this kind of pollination, hydrophily, water pollinated these kind of plants. So B option here is correct. Another question, continued self-pollination results in biofortification. Obviously, students, when we are talking about the biofortification, what we are having? We are having the varieties, production of varieties which will be having high amount of minerals, vitamins, that will be having fortified minerals, vitamins. So this is not the correct option here. Biomagnification, as we have seen in the case of DDT. Inbreeding depression. So continued self-pollination. See here, this is the example of animals. We are here not talking about the pollination as in the case of plants, but related to it. This is also self-fertilization here. And we are having here, see, this was lethal gene, we can say, small a. We are having this combination. Again, this was outbreeding. But again, if we are having the inbreeding, what we are having the combination here? We are having these lethal genes. They have been accumulated in one organism, and that will be very harmful for that organism. So we can say that it leads to inbreeding depression. We have taken here the example of animal, but in plants also, in the living organisms also, this self-pollination can lead to inbreeding depression. That is loss in vigor, fertility. We can say that C option here is correct. If we talk about the heterosis, why it is incorrect? Because heterosis means that the recombinant we will be having, it will be having very high quality. F1 hybrid will be superior to both the parents it was having. Obviously, heterosis is not the correct option. This self-pollination can lead to loss in vigor and fertility that we will be calling as inbreeding depression here. Another question, select the mismatched pair. Is egg apparatus, that is 3-celled. If you can see this image of embryo sac here of a flowering plant here, we are having synergids and eggs. This we are calling as egg apparatus. So obviously it is 3-celled here. Synergids are 2 and egg will be 1 here. This is the correct match here. Synergids, filiform apparatus. These synergids, they are having the filiform apparatus here. So this is also the correct match. Orchids, they are having single ovule in their ovary. They are not having the single ovule in their ovary. They are having multiple ovules. This is the famous example, this orchid, where we will be having multiple ovules in an ovary. Andy is embryo sac, also known as female gametophyte. As you can see in this image, this is the figure of the female gametophyte, also known as embryo sac. So this is also the correct match. We are having the correct option as orchids, which is having multiple ovules in an ovary. Another question, group of morphologically similar. So they are appearing similar in their morphology here, and they are genetically also same. Individuals developed during asexual reproduction. What they are known as sibling. So if we talk about the asexual reproduction, siblings are result of that? Obviously not. Clones, morphologically similar and genetically also same. So this is the correct option here. Gamule cyst. Obviously these are the coverings here. This cyst. This is not the correct option here. B is the correct option. Morphologically and genetically they are identical. That is why they are known as clones and result of asexual reproduction here. Correct option here is B. Another question, choose odd one out with respect to sex determination in honeybees. A, based on number of sets of chromosomes and individual disease. So obviously in the bees we are having this system, the haplodiploid system, and it is based on number of sets of the chromosomes they will be having. The male and female, they will be decided on that. B, fertilized egg develop drones by means of parthenogenesis. Parthenogenesis, when we talk here about unfertilized eggs, not fertilized eggs. So obviously this option here is incorrect, means the statement here is incorrect. Males have half the number of chromosomes as that of the female. So we can say that female, it is having 32, whereas the male, it is having 16. So this statement here is also correct. D is also known as haplodiploid sex determination system. As we have seen, obviously the haploid is male, diploid is female. That is why we are having this term of sex determination in the case of honeybees. So we are having the correct option as B, where we will be having the production of the drones because of the unfertilized eggs by the process of parthenogenesis. So correct option here is B. Another question here, according to scientist Friedrich Miescher, nuclein was the term which was to describe. Now this nuclein is known as DNA. A, acidic substance. If we talk about the DNA, it is phosphorus rich acidic substance. Obviously this term nuclein is for that. A is correct here, B is DNA. This is also correct. C is basic substance. Obviously not. When we are talking about the DNA, RNAs, they are nucleic acids. Obviously they will be acidic substances here. They cannot be basic here. So we are having correct option here as D. All, obviously they are remaining here, A and B, they are correct except C. So D option here is correct. Another question, Taylor conducted the experiments to prove semi-conservative mode of replication of DNA. He conducted this experiment on which organism? Vinca roseaea, Vicia faber, Drosophila melanogaster, and E. coli. When Taylor conducted this experiment to show this mode of DNA replication. To show this mode of DNA replication. As you can see here, we are having 3 modes that was suggested. Dispersive kind of DNA replication, that was not accepted. We are having this conservative mode of replication, that was also not accepted. But we are having certain experiment as was done by the Taylor and one of the scientists also. So they proved that DNA replicates semi-conservatively. Here we are having 1 strand new and 1 strand will be the parental strand. So this was conducted on the Vicia faber, which is a bean. So B option here is correct. Another question, habitat loss and fragmentation. Habitat loss, fragmentation, overexploitation, species invasion, and co-extinction. These are some of the causes of what? Natality, population explosion, competition, and biodiversity loss. Obviously, they are the causes of biodiversity loss. One of the main 4 causes, we are also calling it as evil quartet. And this has led to biodiversity loss. So these are some of the causes or the examples we are having which has led to biodiversity loss. So the D option here is correct. Another question, exotype of sex determination can be seen in XO. Monkey, Drosophila, birds, and grasshopper. So if we have the example of grasshopper, we are having here the female and male. So this exo kind which will be present in the male, we have seen in the earlier question also, it will be present in D, grasshopper, this kind of sex determination here. Another question, that is the last one here, it is saying that if there are 999 bases in an RNA that codes for a protein with 333 amino acids. And now we are having the condition. First of all, we will try to understand, we were having 999 bases here of the RNA. So we know that our codon is triplet. Because of that, we are having the formation of 333 amino acids. Now after that, what is the condition? The base at the position of 901, it was removed. After that, it is deleted such that the length of the RNA becomes now 998 bases. How many codons will be altered? So one mutation it has occurred and it has removed 1 base here at the 901. So earlier what we were having? We are having these 999 bases, and because of that we will get 333 amino acids here. 1 base it has been removed. Now if we talk about here, one it has been removed. Now out of that what we will be having? We are having 900 now. So 99 we will be getting. These will be the bases here. We are talking here now about only 99 because 1 base it has been removed, and because of that we are counting for 900. After that 1 mutation has occurred which has removed 1 base. 99 bases here, and we are having triplet codon here. If we divide by this, 99 divided by 3, what will we be getting? 33 amino acids here. So these 33 amino acids will be altered here. Why? Because 1 was removed, and because of that we are having effect on these 99 bases. So 33 amino acids here, it will be removed. We are having this correct option here as 33. these codons, they will be removed here. So this was all about the 50 questions we were having in the botany. This was all today. Get ready for the mega test too. Thank you.
Hello everyone, welcome to Extramarks NEET Mega Test 1 test discussion. And guys, today you know about the exam, right? AIRTS, all this general information have been shared with you, and I am your physics faculty, and today we'll be discussing the physics paper. Alright guys, so again I'll reiterate all the information about AIRTS. AIRTS, that is All India Rankers test series. So what is the purpose of this? The purpose is to help you get a good rank in the NEET exam and to help you prepare for the NEET exam. All of these paper, okay, so of course the syllabus is complete NEET syllabus, and this is mega test 1. What is mega test 1? That also I'll tell you, and all the related information about NEET AIRTS by Extramarks, that you can get in the description of this video. So after the recording and after this video, feel free to go through the link and look at all the details. But here I'll tell you the general information. So guys, this Extramarks AIRTS that was started on 28th of November. Okay, so what of course the syllabus is complete, 11th, 12th, and all the syllabus for the NEET, whatever is there in the NEET, that is there in AIRTS. Okay, all the subjects, everything. So number of tests, if you look at the number of tests, so 4 minor tests plus 2 mega tests plus 6 full course tests. So there will be 12 tests that will help you prepare for the exam, and this was the 5th test. We are done with 4 minor tests. I'll also show you the schedule of the test. So we are done with 4 minor tests. This is the mega test 1, and then after this we'll have the mega test 2. What is the syllabus of each test? That also I'll tell you. So test pattern is of course NEET based, exactly how it is there in the NEET. So that is what we follow, because of course the purpose is to help you get prepared for the NEET. Alright guys, so this is the schedule. So minor test 1, 2, 3, 4, these are done. These were the partial syllabus test, in which the syllabus was basically, in the minor test 1 it was half of 11th class syllabus, minor 2 was half of second half of the 11th class, and then 3 and 4 were the 12th class. And then we have mega test 1, which is what we are going to discuss today, which was conducted day before yesterday, as you can see here. And then we'll have mega test 2, which will be after 2 weeks. So that will be From now, it is like almost 12 days, so it will be there on 6th of February. And then we have the full test. So guys, in Mega Test 1, it was the complete syllabus of 12th class. Alright, so that was Mega Test 1. In Mega Test 2, we'll have the complete syllabus of 11th class. Alright, and then full test will have the full syllabus, both 11th and 12th, from chapter 1 to last. Alright, so here we are going to discuss Mega Test 1, which will be complete syllabus of 12th class. All the questions, all the 50 questions in physics were from the 12th class syllabus only, from electrostatic to, then we have all the topics of electrostatic, and then we have the optics, and then we have the modern physics. So all of this was covered. Okay guys, so let's begin. And Mega Test 2, it is there on 6, so you have 12 days to prepare, and the syllabus will be complete 11th class syllabus. So go through that, revise all the important formulae, and let's see how do you do in Mega Test 2. But here, of course, Mega Test 1, that is what we are going to discuss. So if you have given AIRTS, great, I'll tell you how to solve the question, and if not, then still this session can be helpful for you, because you can look at the question and then you can see how do we solve this. Of course guys, here this is just test discussion. I'll tell you the solution. I won't teach you the concepts from basics, but I'll tell you the basic formula that is required in each question. Of course, if there is a calculation, that also I won't do exact calculation, but I'll tell you how to solve it. Alright guys, let's begin. Okay, first one was doesn't require any calculation, directly memory based. So it is about Huygens wavefronts, right? Huygens, we know the theory of wavefront. And so what was the theory about? So the theory tried to help explain the concepts related to light, and those concepts were reflection, refraction, and diffraction. All of these were explained by Huygens wavefront theory. So A, B, C, all three were explained. The origin of the spectra, the spectrum that we saw in black body radiation and all that, was not explained by Huygens wavefront theory. So that is why we required the theory of waves, and then we started modern physics from black body radiation. So all the spectra was not basically, in the hydrogen atom we have a spectra, so that cannot be explained by the Huygens wavefront. Alright, so the answer would be D. Okay guys, so next one. This was simple. Next one. So this is about intensity of light. So this is about the interference of light, wave optics. Okay, so what it is about? So it is about two beams of light. The intensities are different. If you remember guys, YDSC, Young double slit experiment, so there mostly the intensities are same, but here the intensities are different. It is I and 4I. Okay, now they basically produce fringe pattern. Phase difference is pi by 2. Pi is of course 180, so this is 90 degree at point A and 180 degree at point B. Then they are asking the difference in the resultant intensity. So guys, this is about resultant intensity, which is caused by the interference of two individual light waves. So what is the formula of resultant intensity? That we have to remember directly. That is very important. That is based on the general idea of superposition of waves. So guys, the formula here for the intensity, resultant intensity, that is given by I1, okay, plus I2, but this is a superimposition vector, so the final result will be 2 root I1, I2, and then we have cos theta. So this is the formula, resultant intensity formula, and that is what we need to apply. So I1 is I, I2 is 4I. So this will be 5I, okay, plus 2 root I1, I2. I1 is I and this is 4I, so this will be 4I squared. So this will be 2 into 2, 4I into cos theta. Right, so that is what we have. So now we just need to put the value of theta, pi by 2. Of course, guys, pi by 2, 90. So cos 90 is 0. So this at A, it will be 5i. And what about the B? So B cos pi. Cos pi is of course minus 1. So at B, it will be 5i minus 4i. So at B, it will be I only. So 5i, and this is I. What is the difference? The difference will be 4i. So that is the answer. So this basically the whole question was from this formula, I1 plus I2 plus 2 root I1, I2 cos. If you remember this, you can solve it. If you don't remember, doesn't matter how much time you spend here, you won't be able to solve it. So guys, important formula in wave optics. So this you have to remember. Alright. Next one. Okay, so this is about fringe width. Again, simple question if you remember directly the formula of a fringe width. So what is the fringe width? Let me write the formula directly. So that is d lambda by d. Okay, that is the fringe width. D is the distance between the screen and the slits, and lambda is of course wavelength, and D is the distance between the slits, right? So now what they are saying that fringe width is 2.4 when red light is used. What is the fringe width when how much it will change when blue light is used. So the only difference is lambda d, capital D, small d are same. So you can put lambda given for the red light and then see the fringe width, and then you can put the lambda for the blue light and then see the fringe width. So if you see guys, if we compare the lambda, so this is 6400, this is 4000. So what is the change here? So basically 6400 and 4000, if we talk about the ratio, so this is basically, you can say this is 10, this is 40, this is 64. So this is 10, this is 16, this is 5, this is 8. So the ratio is 5:8. So same will be the ratio in the fringe width because directly proportional. So if this way, so the fringe width will become 5/8. So this is 2.4 into 5/8. So that will be 1.5. So the fringe will be 1.5. So what will be the difference? The difference will be 0.9, right? So that is a very intuitive way to solve it. Of course, in an exam, you can do the exact calculation and get the answer. All right. Next one. So again, guys, this is about intensity, but here they are talking about the theta. This theta value they are asking. So actually, this is about the path difference. When there are two slits, what is the path difference? So the path difference is basically we need to draw a perpendicular here. Okay, this is a perpendicular, so this is the path difference. And what is the, so if this, let's say the width of the two slits, basically the distance between the two slits is here 2 lambda. So this is, we can just, so this is perpendicular, so I can just use cos theta to find this width. Basically I want to find this width. That is the path difference between them. Okay, so that will be 2 lambda cos theta. Okay, so that is what I want to, from here I'll get theta. Okay, how? So this is the path difference, 2 lambda cos theta, and here guys they have given the resultant intensity is 3 I naught by 4, where I naught is the maximum intensity, which is, let's say, though here the intensity will be I naught. So guys, we need to remember that at any point in general, the formula of intensity is given by it is I naught cos square, you can use the symbol phi by 2. Okay, phi, so this I naught is the maximum intensity. Okay, cos square phi by 2. What is phi? So guys, phi is the phase difference between the two waves. Okay, so here this is given as 3 I naught by 4. So this will be 3 by 4 is cos square phi by 2. And if we talk about cos phi by 2, so that will be cos phi by 2. That will be root 3 by 2. And root 3 by 2, guys, we know cos, this will be cos 60 degree or cos 30 degree because cos 30 is root 3 by 2. So phi by 2 is 30. So what is phi? Phi will be 60 degree. So that is the thing. Phi is 60 degree. And guys, this is the phase difference. And we have to convert this phase difference into path difference, because we have the path difference 2 lambda cos theta. So we have to convert this into path difference. So guys, if we know phase difference, can we convert into path difference? Yes, the relation is path difference is given by 2 pi by lambda into the or lambda by 2 pi actually. So this will be lambda by 2 pi into the phase difference. So this will give you the path difference. So lambda by 2 pi into, so this 60, which is pi by 3. So pi and pi will get cancelled. So this will come out to be lambda by 6. Right, lambda by 6. So this is your path difference. So we'll compare 2 lambda cos theta equals to lambda by 6. So there you'll get the value of cos theta, which will be 1 by 12. So theta will be cos inverse 1 by 12. So that is the answer. So this was a little bit complicated. We required the knowledge of intensity as well as how to find path difference from the phase difference. And then we also needed to know from the diagram that 2 lambda cos theta will be the path difference, and then we'll compare. Alright, let's continue. Next one. This is, guys, disc is placed on the surface of pond. So let's say this is the disc, and then we have a pond of water. Okay, source of light is placed 4 meters below. So this is the source of light, let's say. So the minimum radius of disc, so that light is not coming out. So there okay, let's say this is the disc little bit. So light is not coming out. Why will light not come out? Because, guys, so let's say the light goes like this. Okay, light goes like this. It will not come out because there will be TIR. That is the thing. So there will be, because the light is trying to go from water to air, so denser to rarer, so TIR can possible. So basically what they are asking, what should be the minimum radius so that there will be TIR. So if there is TIR, basically guys, we know TIR, so the angle. So they are talking about the minimum radius, so it means we have to take the boundary case, which means this will be the critical angle, right? At what is happening here is basically the so if I draw the normal, so this is my critical angle, this is the critical angle. So now we can find easily, so this is critical angle and we know, so this is a refractive index is 5/3, so we can find this in terms of radius and depth, and then we can also find the angle in terms of critical angle. So as we know from the laws of refraction, the Snell's law, that sine theta c, which is the critical angle, is 1 by the refractive index, so that is be 1/3, so this will be 3/5. So sine theta c here, that is 3/5 from the knowledge of Snell's law. And if you look at the diagram guys here, so this is depth is given, which is 4, so and they are talking about the radius. So if I use tan theta c here, so tan theta c, that is going to be r/4, right? And what is tan theta c? So if I know sin theta c, I can find tan theta c also. I just need to draw the diagram and I'll get the value. So if I know sin theta c, then tan theta c in that case will be 3/4, right? Because let's draw a hypothetical triangle. So sin theta c is 3/5, so it is perpendicular is 3, hypotenuse is 5. So in this same triangle, what is tan theta? Tan theta is perpendicular upon base. So guys, if this is 3, 5, this is 4, so tan theta c will be 3/4. So now if we compare, r/4 is 3/4, so what is r? R is 3. So that is the okay, so yeah, so that will be the answer accordingly. I think they are talking about the if the answer should be 3 if they are talking about the radius, but if they are asking about the diameter, then it will be 6. Alright, let's continue. Okay, so this is now from reflection, and guys, these type of question, they look complicated, but things become easier once we draw the diagram and we use the laws of reflection. Alright, so basically guys, what is happening here, there is a reflection happening at two surfaces. So let's draw the diagram in a little bit more detail. So this is the first reflection, and then light will go like this, and this will be the second reflection. So this is kind of the diagram that is given to us, and what is happening is this is theta, so of course guys, this will also be theta. So now we need to draw some normals here, because we want to find angle of incidence and reflection. So this is the angle of this is the normal for the first mirror M1. So let's say guys, this we can take we call it, let's say, alpha. So this will also be alpha, right? This is alpha, so this is alpha. Similarly, what I'll do is I'll draw the normal here also. Okay, let's call this beta. So this will be beta, and if we talk about this angle, so this will also, because by symmetry, this will also be theta. So theta, theta, and if we talk about this one, so that will be if this is alpha, then this will be 90 minus alpha. So this will be 90 minus. So 90 minus alpha plus 2 theta, that is 180, because it is a complete triangle. So here we can find the relation between alpha and theta, and that is going to be 2 theta minus alpha, and that is 90. Alright, so that is the first equation. Basically what they are asking is angle theta. Okay, so this is the first equation. Of course we don't know alpha. Now we have to write the similar kind of equation for basically this using beta also. So that is the thing. So this is basically just about writing the equations of triangle, that our total angle is 180. So using that, and then using the laws of reflection, we can find this. Of course I am not doing the complete calculation here, that is would be time taking, but the concept here is the laws of reflection, angle of incidence is equals to angle of reflection, and then using the properties of triangle, that the total angle is 180, right? Sometimes we also have to use like corresponding angle, like this was theta, this is also theta, so that is little bit of geometry, corresponding angle, alternate interior angle, all of that. But other than that, these questions are kind of simple. For example, here we can take this triangle, and then we can write the relation between 2 beta plus alpha equals to, so here I can write 2 beta plus alpha, that is 90. And guys, we know beta is what? Beta is 90 minus theta. So this 2 beta I can write 2 into 90 minus theta. So this way I can write another equation. So basically we can make more equation until we get the equal number of equation as the equal number of variable. We can make equation and then solve it. and we'll get the answer. Alright, so I'll leave it up to you guys. If you solve it, you'll get theta as 60 degree. Okay, let's continue. Okay, this is about astronomical refractive telescope. Okay, and it is saying objective has focal length this, eyepiece is this, what is the magnification and also length. Of course, guys, the image form is inverted, that you remember. In refractive telescope it is inverted. C must be correct. Length, of course, is, guys, the addition of focal length. If you remember the structure of this telescope, so it is the focal length of the objective plus the focal length of the eyepiece. So 20 meter plus 2 centimeter. 2 centimeter is 0.02 meter. So 20 plus 0.02. So 20.02. And magnification, guys, if you remember the formula of magnification in case of this the refractive type telescope is minus FO by FE. Right? Minus is because inverted FO, objective focal length, divided by the eyepiece. So if you put the value, so this is 20, this is 0.02. So 20 divided by 0.02, that is going to be 1000, and minus, so minus 1000, that is your magnification. They're talking about here magnitude only, so magnitude is 1000, and minus means inverted. So A, B, C, all three are correct. That is why answer is D. Alright. Okay, so this is about prism, and ray of light is incident at 60, angle of prism is given, emergent ray makes 30 degree with the incident ray. What is the refractive index of a prism? So this is little bit of okay, so if we draw the prism here, and this is angle of prism, and let's say the light goes like this, and then it goes like this, and then like this. Okay, kind of like this. And these angle, guys, we know some of the properties. So this is if I draw the normal, so this is my R1, and if I draw the normal here also, this is R2. We know some of the property that A equals to R1 plus R2, and there is one more very important formula here, and that is A plus del equals to I plus E, or A plus del is I plus E. So A del is what? Deviation. I incident angle, E emergent angle. So now here what is given? This incident I is given 60, angle of prism is given 30, this deviation is also given, that is 30, which is the angle between incident and the final ray emergent ray. So here we can find the emergent angle. So let's try to find it. So A is what? 30 plus deviation is also 30 equals to I, which is 60, plus E. So here you can see, guys, emergent angle is 0. So it means finally the light ray is normal. Basically, if I draw the final ray here, it is normal. That is why final angle is 0. So the ray, if I draw the final ray here, that will also basically along the normal. So it is going like this somehow. Alright. So now they are asking what is the reference. So guys, here if this emergent ray is angle is 0, so of course this R2 is also 0, because if let's say R is 0, then I must also be 0, right? So if the emergent ray is along the normal, so this ray, which is basically going on this surface of the prism, that must also be along the normal. So this R2 is also 0. If this R2 is 0, that means A equals to R1. So if we talk about R1, that is A, and that is 30 degree. So using that, now we know, I know I, okay, I, if I draw the normal here, so this is, so I is also given in the question, which is 60, and I know sine, this is Snell's law, so sine I by sine R, that will be your the refractive index. So that is what they are asking. Sine I we know, sine R we know, just put the values, you'll get n, and that is going to be root 3. So that is your answer, 1.73, which is the value of root 3. Alright. Okay, so this is about focal length of combination. So two identical glass equiconvex lens. So these are two lenses which are put in contact. Okay, kind of like this. The space between them is filled with water. So guys, in between them we have water. Let me draw it a little bit more accurately. Okay, so these are the let's say two convex lens. In between them we have water. So if I draw the water also here. So this is the kind of system that we have. What is the focal length of the combination? So guys, this is actually this water thing that is there in between. If we separate, like if we look at it independently, so it is like this. Basically guys, the gap between two convex lens is basically, it becomes like a concave lens. So this is a concave lens. The water, it basically behaves like a concave lens. So now it is like three lenses, convex, then concave, then convex. So if there are three lenses in contact, what is the power final, it is the addition of individual power. So it will be P1 plus P2 plus P3. Okay, the question is, so P1 we can find easily because so what we can do is, the focal length of this is given. So power of 1 is 1 by f, power of 3 is also 1 by f, power is 1 by f. So but what about the second one, this water one? What is the power of this one? So power is 1 by f. So we need to find the focal length of this one. And how do we find focal length? The Lensmaker formula, very important formula in this chapter. So as using Lensmaker formula, I can find the power of this one. And Lensmaker formula is, if I hope you remember. So guys, Lensmaker formula you can use. So let me write it for you. That is mu water minus 1, and that is going to be 1 by, so radius is same, whatever was the radius of this convex lens, that is going to be the radius of this water concave lens also. So 1 by R1 minus 1 by R2. So R1, R2 both are same. So using that, UW is given 4 by 3. Using that, you need to find 1 by F for the concave lens and then add all these power, and then that will give you the answer. Alright guys, so rest is just calculation. The most important thing was that this inside water will behave as a concave lens, which we can find focal length using the Lensmaker formula. Alright. Okay, next one. Okay, what about this one? So this is again lenses. So this is plano-convex lens, plano-concave lens. Okay, there is a gap also. And they're asking what is the location of the final image. Okay. So little bit more involving. Why? So first we need to find the focal length of this one. How? Lensmaker formula. So let's find the focal length of the first one. So 1 by f equals to mu minus 1. So refractive index is 1.5 minus 1. Okay, 1 by r1. So guys, what is the r1 for this one? So this is straight line, vertical line. So for vertical line, the radius is infinite. So 1 by r will be infinite. Minus 1 by r2. R2 will be negative because the center is on this side. So this will become plus 1 by r. What is the radius? 10. Yeah, radius is given. So this will be your 1 by f, which is 0.5 divided by 10, which is 1 by. So focal length will be 20 centimeters. So guys, this will have a focal length of 20. It means the parallel lights are coming. They will form image here, which will, let's say, V1. But this V1, now it will behave as an object for this second lens. So we need to find, using Lensmaker formula, the focal length of the second lens. So the second lens is plano-concave. So for this one, the R1 will be negative and R2 will be infinite. So we'll use the same formula, but here R1 will be negative. So the focal length of this one, if we solve this, that will be minus 20. Why? Because it is concave, so naturally the focal length will be negative. So now this image will behave as an object for this lens. So now we need to use the lens equation, which is 1 by V, which is the final image, minus 1 by, let's call it V1. And that is 1 by F, which is minus 1 by 20. Using this, we'll get the V, and that will give you the answer. But only thing to take care is here, V1 will not be negative, V1 will be plus, because the V1 is on this side. So if you put V1 as, what is the V1 we're getting? So here V1 will be basically this, we have to measure from. So this will be 10 only, because total, this is the focal length of the first one, plano-convex, so total was 20. So it means parallel rays will meet at 20. So, but from this one, the plano-concave, the distance is only 10. So this V1 will be 10. Alright, and so using that you can find V, and that will give you the answer, which will also be 20 if you solve this. Okay guys, let's continue. Next one, this is kind of simple. So mu1, mu2, mu3, that is given. Mu2 is more mu1 than mu3. So here it is the denser liquid, okay, compared to lens. Here it is the rarer liquid. So this is a, what kind of lens is this? Is a convex lens. We know it is a converging lens. But if the medium is more dense than the lens, then it becomes a diverging lens. So basically the rays above, they will diverge. Okay, so the above part of this lens is diverging. Below is like normal only, just like you can imagine it is air, because air is less dense. So below part is converging. So one part is diverging, one part is converging. So we'll get two different light beams. Basically we get a we'll get a one convergent beam and one divergent. Both will get. Above one will produce diverging, below one will produce converging. Okay. So this was this was the 11th one. Let's go to the Well, again guys, this is okay, this is first kind of question about apparent depth. Remember the formula of apparent depth, very simple. Actual depth divided by mu. That is your apparent depth. That's it. So if you use this, you'll get the answer. H by 3 mu1 plus H by 3 mu2 plus H by 3 mu3. So H by 3 will be common, 1 by mu1 plus 1 by mu2 plus 1 by mu3. Very simple. Okay, critical angle they are asking. So guys, we need to remember basically Snell's law. So n1 sine I equals to n2. That will give you, because sine r is 90, so this will give you the critical angle. So first critical angle, glass is theta 1. So if you talk about glass, so sine theta 1 equals to n2. Basically for glass it is here, so 1 by mu g, which is 2 by 3. So so this is sine theta 1 is 2 by 3. Similarly sine theta 2 for water, that is going to be 3 by 4. Now they are asking between the glass and the water. So light will go from glass to water, rarer to denser. So what will be the so you can use Lenz's law, very simple. N1, which is 3 by 2 into sine, let's say theta 3, equals to the refractive index of the water, which is 4 by 3. So what is this going to be? This is going to be 8 by 9. So guys, now we can compare. So as we know, sine theta is more, it means theta is more. So this is 2 by 3, this is 3 by 4, this is 8 by, this is largest, right? Then this, then this. So theta, this is theta 3, which is what they are asking. So theta 3 will be the greatest, then theta 2, and then theta 1. So that is why the answer will be greater than theta 2. It is greater than theta 1 also, and theta 2 also. Alright. Okay, next one based directly memory based. So you can say that when we go from basically gamma rays, they have the most frequency and they have the least wavelength. So it is question about decreasing, so that is why it is the answer. Okay, this is again guys memory based. This is from dual nature of light, we know, or it is also from electromagnetic wave, that we know a wave has both momentum and energy, right? When it strikes a wall, the light beams, they produce force. We know the formula of force also, but here they have momentum and they have energy. Okay, this is about transformer, very simple one. They are trying to confuse us by giving so much information, but what they are asking is the input power. So as we know that power, whatever is the input power, that is going to be output power, because power cannot be produced or destroyed, conservation of energy, but it is 60% efficient. So it means 40% of the energy it is wasting into heat or something. So if output power is basically 60% of input power. So output power is basically 60% of input power. So what is the output power? The output power is 9 kilowatt. So this is 9 kilowatt. Okay, so 9 kilowatt, that is 60%, which means 60 by 100 into P input. So using this we can find P input, and that is going to be 15. Very simple. All of this 11,220, that was just to confuse you, that is not required. Okay, next one, reading of voltmeter. So they are asking what is the potential difference around the resistor. They have given this 100, this 100, total is 200. So guys, we know it is a RLC circuit, and in RLC circuit the potential combined for this is given by, if you remember from the phasor diagram also remember, so the V is given by VR square plus VL minus VC. This is VL minus VC whole square. That is the formula of the resultant potential difference or potential, right? Because L and C, they are in opposite phase, and that phase is compared to a perpendicular compared to the phase of the resistance. So that is why we have this formula. So now VL and VC both are 100. So if we compare, so this is what they are asking, V square plus, this is 100 minus 100 whole square, which is 0. So this is V square, and total is 200, that is given. This is 200. So 200 is V square. So under root of V square, which is basically VR. So VR will be 200. So that is the answer. Very simple. So we need to remember this important formula for RLC circuit, the resultant potential. So V, which is 200, that is VR square plus VL minus VC whole square. Okay, total energy stored. So guys, this is LC circuit. We need to know the formula of total energy stored, and that is initially. So initially, all the energy will be stored basically in the capacitor. There will be no energy in the this inductor. So in the steady state, maybe it will be different, but total energy stored initially, we need to remember the formula, that's Q naught square by 2C, right? That is the energy of a capacitor. So Q is given, everything is given, and we just need to put the values, right? So Q is 5 milli, and this 2C is also given. So very simple, just put the values, get the answer. Next one, so mutual inductance, guys. This is mutual, so this is what it is, a solenoid, and inside solenoid what do we have? We have a circular coil, and they are asking the mutual inductance. So guys, we need to follow the formula of mutual inductance, and that is going to be the magnetic field, or due to the solenoid, is mu naught Ni. So I will not be there because mutual inductance will be, we have to divide by current. So mu naught N into the area of this circular coil, and that is A2, which is the area, but we have to also multiply by the number of tons, because around each ton this is the mutual inductance, but if there are N2 tons, then we have to multiply by N2. So this is your final formula. N is the number of tons per unit length for the solenoid. That is important, because mu naught Ni, that is the magnetic field of solenoid, which is N is the number of tons per unit length. So now we just need to put the values, we'll get the answer. So mu naught, we need to put the values of mu naught also, and that is going to be 4 pi into 10 power minus 7. Then N, what is N? 15 tons per centimeter. Okay, so if we convert it into meter, so that is going to be, or we can keep it in the centimeter also, that is fine, because all the values are in centimeter, so they will get cancelled anyway. Only thing we need to take care is the, yeah, so everything is given. So A2, guys, is given, and that is the circular, the cross section of the circular, that is 4 centimeter square, and this we can keep it in centimeter, not an issue. So this A2 will be 4, and then this N2, so what is the N2? It has 10 tons, so that is your N2. So this is your mutual inductance, right? So this, we'll put the values and we'll get the answer. Very simple. So 4 pi, you can put the values here and you'll get the answer. Alright, so let's continue. Okay, next one. So this is, they are asking, if we move the magnet, what will be the direction of the induced current? So guys, we know this is from Lenz's law, right? What will be the induced current if we move, if we basically change the magnetic field? So here, if you can see, let's say we take it towards A. So if we are we are taking it like this. So south pole is basically going inside this part. So guys, we know inside the magnetic field of a bar magnet is from north to south, right? So basically the magnetic field are like this. So magnetic field inside, will be like this. So these field which are going from left to right inside this A solenoid, let's say, or A coil, that is increasing. So the A will try to oppose it. That is the Lenz's law, that it will try to oppose the change in the flux. So if flux is increasing in this direction towards right, the current will be such that it will oppose this one. So magnetic field due to the current in loop A will be towards left. So what will be the direction of the current? So right hand thumb rule, if the magnetic field is towards left, the current will be like this, right? So what will be the direction of the current? So basically the current is going, basically if it is going like this. So what kind of direction is this? It is towards left. So this is basically the anti-clockwise direction. So in A, the current will be anti-clockwise, right? And anti-clockwise, of course, means from Q to P, right? In the coil, it will be anti-clockwise, but if we talk about these wire, so then it will be clockwise, because it is going from Q to P, and then from on the coil it is anti-clockwise. So if we talk about the wire, it will be from Q to P, and similarly here, if here guys, for the B coil, the north pole is going away, and we know that the magnetic field from north is basically like this. So these fields are decreasing. So B coil will try to increase these fields. So the magnetic field due to the B coil inside will be towards right. So what is the direction of the current? So is the direction of current is like this. So that is your clockwise direction in this. So clockwise inside the coil, but if you talk about the wire, it will be from X to Y, right? Which is like this. So that is the thing. So basically it is Lenz's law. Rest is just, little bit of complication, but if you follow Lenz's law correctly, then you will reach the final answer. Alright. Okay. This one, okay, a lot of things are happening here. What is this? So this is a wire moving through magnetic field, and then it is connected with this circuit, and there is resistance also. They are asking the force needed to move the rod. So first thing, if we are moving the rod, there will be induced EMF. What will be the value of induced EMF? So you know the formula, and that is going to be BVL. V is the velocity, L is the length. So that is your induced EMF. Now we can find the current. Why are we finding current? So we'll see. So EMF divided by the resistance. So that is a current. So there will be this current in the circuit. Right? There will be current in the circuit through the resistor and through the wire also. So now it is like a current carrying wire moving in a magnetic field. So we know when a wire moves in a magnetic field, there will be force on the wire, and what is the force? And that is BIL. So that is the force. So B is given, I we have found, which is BVL by R into L. So if we put everything, we'll get the final answer, and that is this force. At BIL, what is I? EMF by R, what is EMF? BVL. So everything is given, we just need to put the values and get the answer. Alright. Okay. Magnetic flux. Very simple. Magnetic flux is what? Magnetic field into the area, right? But which area? Perpendicular area. Okay. So square lies in the XY plane. So let's say this is your X. This is your Y. So the square is like lying in this. Magnetic field is given. Flux. So guys, flux. Here if we talk about the flux, it means the magnetic field must be towards Z axis, because the flux is measured when the magnetic field is like perpendicular to the surface, or it is in the direction of area vector. Area vector for this one is along Z axis. So here if we talk about the flux, that is going to be magnetic field which is on the Z axis, which is 5. Okay, into area, what is going to be the area? So X, so area will be X square. So 5 into X square, that is your area, times the magnetic field, which is 5 into B. B naught actually here. So 5 into B naught. So 5 into B naught, 5 into B naught into X square, that will be your flux. Alright. So option A. Okay, toroid. So this is about toroid, and they're asking relative permittivity. So guys, we need to remember the magnetic field of a toroid. That is kind of like similar to solenoid only. That is mu naught, but here it is relative permittivity. So there is maybe another material. So mu naught into mu r into N into I. What is N? N is again number of, basically it is number of turns per unit length. So let's put the values. Mu naught, okay, into mu r into number of turns is 400, but per unit length, what is the total length? Okay, total length is 40 centimeters, so it means 0.4. Okay, into I, which is 2. So that is our magnetic field, which is 1. Right, so everything is given, we need to find mu r. Okay, so this will get cancelled, this will become 1000. 4, this is 1000. So mu r is basically 1 by 2000 mu naught. Alright, and so here we need to put the values of mu naught, and we'll get the answer. Alright guys, so let's continue. Okay, next one. This is a wire which is connecting wire ABC. Mass is given, current is given. What is the acceleration of the wire? So acceleration, of course, is force upon mass. Mass is given. We just need to find the force. So if we talk about the force, so guys, we know that we can use the Fleming's left hand rule or this right hand palm rule, which we can find to find force. So the current here is this, magnetic field is this. So on this part AB, the force is in this direction, right? And if we talk about this part, the force is in this direction. So now if we find the resultant, so the horizontal component will get cancelled, and finally the force will be only the vertical direction, right? So the force, resultant force due to these two will be vertical, right? Because horizontal will get cancelled. So the force will be along y-axis, that is for sure. But all the options are also saying that. So we need to find magnitude. So as magnitude is, this horizontal component will get cancelled. So we need to, so force due to this one. And if we talk about the hypotenuse basically we can divide into two parts. One is the vertical part, other is the horizontal part. So the vertical part of the hypotenuse, which will be this, so these two will get, the force will get cancelled. Only the horizontal part of the hypotenuse, which is AC, that will give you the force. So the force we know is given by BIL, but L we have to take for the AC, the length of AC. So everything is given, just find the length of AC, which will be 3. B is given, I is also given, you will get the answer. Divide by the mass, this is of course force, and if you divide it by the mass, that will give you the acceleration. So little bit of geometry is involved. The important thing to realize was that the force is BIL, L we have to take only for AC because the other component will get cancelled. Alright. Okay, this is from paramagnetic material. So guys, we know this is MH curve. So guys, M for magnetic material, it is directly proportional to H. This is memory based. So M is directly proportional. There is some constant divided by temperature is also there into H. So yeah, that is the relation between M and H. So directly proportional to H, that is why we have a straight line. Okay, independent, again memory based, vertical component of earth field is not used to specify the earth magnetic field. We talk about horizontal and then we talk about these two, but this is not used to talk about the earth magnetic field. Okay, this one, so this is about magnetic moment. Magnetic moment is what, guys? Magnetic moment is I into A into N, the number of tons, that is your magnetic moment. So yeah, we just need to put the value. So here the total length is 18A, but here we can only see 6A. So it means there must be 3 tons, right? So N is 3. I is current. So guys, current is what? V by R. So V naught by R into the area. So guys, what is the area of this one? So this you can find actually using geometry. I'll write it directly here. So this is 3 root 3 by 2 A square. That is your area. You can find it very easily. You can find this one triangle and then multiply by 6. That also you can do. Okay, let's continue. Yeah. So this is about galvanometer, and they are giving the shunt and they are asking what is the maximum current that can be measured. So guys, we need to remember the current measured by ammeter in terms of the shunt and the galvanometer current, that is Ig 1 plus the galvanometer current Rg divided by the shunt. That is the formula that basically tells us if Ig is given, what is Ia, which will of course always be greater than Ig. So here if you just put the values, you will get the answer. Ig is what? 10 milliampere. 1 plus Rg, which is 40 divided by S, which is 2. So 1 plus 20, 21. 21 into Ig. So this will be in ampere, it will be 0.21. Alright. Proton, alpha particle radius. So guys, if we talk about the radius, what about the radius? So in a magnetic field, we know that magnetic field is basically equals to centripetal force. So mv square by r equals to q, vb, we get cancelled, and r we get is mv by qb, right? But here they are talking about energy. So in terms of energy, the r can be given by root 2 m kinetic energy divided by qb. This is your radius. So here kinetic energy is same, and of course b is also same, 2 is same. So it is proportional to root m divided by the charge. So we have to see what is the value of root m by q for all of these, and that will give you us the value. So for proton, the mass, this is 1 hydrogen atom, so we can take mass for proton, that is 1. For deuteron it is 2, right? Isotope of it is hydrogen, and alpha particle mass is 4, right? 2 protons, 2 neutrons. And if we talk about the charge, so proton charge is if we talk about the charge proton, let's say q proton, it is E, and deuteron it is the charge is also same, but on alpha particle the charge is double, 2e, right? Because it is 2 protons, so 2e. So these are the charges. So now we'll just look at the ratio and we'll get the answer, right? So it is 1 by 1, it is root 2 by 1, and here it is 4 by 2, which is root 2. So yeah, so this is under root, so under root of 4 is 2, so this is 2 by 2, 1. So this is also 1, but this is root 2 and this is also 1. So answer is 1 ratio root 2 ratio 1, which is option C. Alright, so just follow the formula, write it in terms of kinetic energy, you'll get the answer. Okay, frequency, guys, we know frequency, which is basically the related to time period, that is not related to radius. You can find the formula also. So using this, we found the formula mv by qb. So frequency, or we can find time period, that is 2 by r divided by p. So r will get cancelled. Time period of frequency does not depend on the radius. So frequency is doubled, radius will not change. Okay guys, next one. So this is about potentiometer, and they're talking about this L length. So guys, this battery has internal this resistance also, which is connected to the battery, that is 10. So now if we talk about the current in the circuit first. So guys, the current in the potentiometer wire, that is V by total resistance. So guys, V and total resistance is 10 plus the resistance of the potentiometer wire, and the resistance of the potentiometer wire is 20. So total potential of the battery divided by 10 plus 20. 10 why? Because this 10 is also there. So V is what? 6. So 6 divided by 30, that is 1 by 5. So that is your current. Now if you talk about the potentiometer wire, guys, the resistance is 20, and so potential difference across the wire, which we can say VA, from VA to B, that is going to be IR. So 1 by 5 into the 20, so that is going to be 4 volt. So basically it is the 4 volt that is getting divided from A to B. Now they are asking what is the length in which we will get 2.4. So we know it is directly proportional to length, because more is the length, more is the resistance, more will be the potential difference across. So it means this will be 60%. L will be 60% because total is 4 and we want 2.4, which is 2.4 is 60% of 4. So this length L will be 60% of total length AB. AB is what? 200 centimeters. So 60% of 200 centimeters. So 60% is of course 120. So that is why we get 120. Little bit involving, but if you know potentiometer, you can solve it very easily. Okay, potential if VC minus V, very simple guys. Just find the current. We know 8 ampere is coming. So this above we have 11 ohm resistance, below we have 6. We know current, it will divide in the inverse ratio of resistances. So the current which is going up, let's say out of 8 will be 8 multiply the inverse ratio. So 11 total is, this is 11, this is 6, so 11. If we talk about the current which will go above, so total is 11 divided by, so this is 11, okay, we can draw it like this. This is 11 and this is 6. Okay, and so if this is 8 ampere, so what will be the current that will go here? So 8 divided by 17 into 6. That is the current that will go here. Why into 6? Because it is inversely proportional to the resistance in parallel. So this is the current that will go above. So if this is the current that will go above, we can easily find basically VA. And similarly here, what is the current that will go below? That is going to be 8 by 17 into 11. So if we know this, we can easily find the current in 2 ohm. So this will be the current in 5 and 6. This is the current in 4 and 2. So using this, we can find the potential. Potential is what? I into R. So this potential, let's say you can take this as 0, let's say. So what is the potential across 2? 2 into this. So 16 by 17 into 11. So this potential is 16 by 17 into 11. And what about this one? This is 8 by 17 into 6 into 6. So now just find the difference and you'll get the answer. So if you find the difference between these, you'll get 6.6. That is the answer. Okay, next one. So there's a battery, internal resistance is also there, so which we can draw it, like this, 2. Okay, and this one has 1, so we can draw it like this, 1. Okay, what is the current? So this, now we need to find the current here. So let's say the current is, let's say I, which is what we want to find. So let's say the I1 current is coming here. This is I, this is what? I1 minus I, right? Because I1 is coming, I goes here. What is remaining will be I1 minus I. Okay, so on this branch there will be I1 minus I. This is I, and this is of course I1. So now this is, we just need to use Kirchhoff law. Okay, twice, one in this loop and other is in this loop, and then we'll find the value of I. So this is Kirchhoff law, guys. If you use Kirchhoff law here and here, you'll get the answer. Kirchhoff law, if I try to apply, so it is, okay, I'll tell you how to apply in one loop and then you can apply in the other. So let's talk about the first loop. So if I apply the Kirchhoff law, let's say for the resistance, so that is, I'll start final minus initial, so minus 10 into I, then we get the battery, so plus 7, and then we have the internal resistance, so minus 2 I1, that is 0. So the equation I'm getting is 2 I1 plus 10I, that is 7. That is our first equation, guys. So using Kirchhoff law, I have written first equation. Now write the second one for the second loop. 2 variable, 2 equation. Solve it, get I1, and I will be your answer. So that's it. Okay, next one, R1, R2. Where is R1? This is R1. Where is R2? This is R2. Okay. They have given current here, 20 ohm, 1. So guys, we know these three are in parallel. 10, if you see, these three. 1, 2, 3. These are in parallel, so of course the potential difference is same across. What is the potential difference across this one? IR, so 20 volt. Okay, so it means across R2 also the potential difference must be 20. So 0.5 into R2, that is going to be 20. So 0.5 into R2. Okay, that is going to be 20. So what is R2? R2 will be 40, right? So that is your answer for R2. There's only one option which is saying R2 is 40. So option A, you don't even need to solve R1. But yes, you can solve R1 also, because this is 0.5. Then you have to find current. So in 40 ohm current is 0.5. So in 10 ohm current will be 4 times. So this will be 2 ampere. And in this 20 the current will be 1 ampere. So total current is 0.5 plus 2 plus 1, 3.5. So then you can find R2, because total current is 3.5. And this you can find, of course, the resultant of these, and then compare. Basically it will be 3.5 is 69 upon R equivalent, and R equivalent is R1 plus the parallel resultant of these three. Using that you can find R1. But not needed because of the options given. We can just find R2 using the fact that potential difference will be same in these three which are parallel. Okay. Power, they are asking how much. So what is the relation between power and potential difference? So P equals to V square by R. R is constant for a device, so V square by R. So it is proportional to V square. So if we decrease V, power will decrease. If we decrease by 2.5%, power will decrease by 5%. This is a question from, if you can say, it is kind of using what we studied in 11th class, error and all. So if this is proportional to V square, so if the error in V is 2.5%, error in P will be double. So it is kind of like this. If it decreases by 2.5, it will decrease by 5. That is kind of approximate, which is applicable for small values. If we try to find exactly, it will be not exactly 5. It will be slightly, it will be slightly less than 5 or more than 5. Depends on the values, but approximately we can do it will be 5. Okay, next one. This was the, I would say, the most difficult question of the complete AIRTS, this mega test one. So they are asking what is the equivalent resistance between these two. So this is concentric sphere. Okay, so guys, what we'll do here is we'll take a element. So this is R1. We'll take a element of radius, let's say R. Okay, and the total is of course R2. So if we take an element, and this element will have a very thin, it is a thin concentric shell, thin hollow sphere, you can say. So this, if we talk about this hollow sphere, what is the resistance of this one? So as we know, resistance is rho L by A. So rho, what is the length? So length is basically the thickness here, which is dr. So rho into dr divided by the area of this one. So this is a sphere, so 4 pi r square. So this is the resistance of this one, but of course R is changing. So this is, we can say this is dr. This is dr, small resistance. To find the final equivalent, I need to integrate it. So rho by 4 pi will come outside density, and dr by r square from R1 to R2. So this will give you the answer. So density is given, resistivity actually rho L by A, sorry, not density, resistivity, rho L by A, that is the formula of resistance. So if you integrate, so of course integration will be minus 1 by R. So if you put the limit, so you'll get 1 by R1 minus 1 by R2. So rho by 4 pi, 1 by R1 minus 1 by R2. Take LCM, so you'll get the B option. So this was, we had to take element and then integrate. This is not possible without integration, this question. Okay, minimum number of capacitor 8F 250 volts. So guys, each has this and we want this. So how do, so first we have to solve for potential. In these question, first we have to solve a potential. So 1 is 250. So we will convert in, connect in series, because in series the potential will be divided. So each is 250. So what will be the total potential in series if we connect 4,000? And that is what we need. We need 1000, so we'll connect 4. So 4 are in series, but what is their capacitance? So guys, in series we know capacitance decreases. So it will be 1 by C equals to 1 by 8 plus 1 by 8. So basically the total will be in series, the equivalent resistance will be 2 microfarad. So how many parallel we need to connect? Total we want 16. So this is 2. So in parallel we know it will add. So for 1, 2, so how many we need to connect? 8, because 8 into 2 will be 16. So we need to connect these 8, right? 8 parallel combination of 4 capacitor. So this we have to connect 8. So what will be the number? 4 into 8, 32. That is the answer. Alright. Flux. So this is about tons. So flux. So guys, flux is very simple. It is N each ton. So they are asking about each ton only. So they are not asking the total. So then the flux will be basically I. We know that flux is L into I. That's it. So just multiply the two, get the answer. Here they are giving the values in mu naught by 4 pi, which is 10 to the power minus 7. So that is the answer. If you multiply this, we'll get 10 to the power minus 7. Okay. Next one. So this is again, this is about the charge. So this is from which one? Gauss laws. They are asking the charge within the Q. Flux is given. So it's very simple. Flux is given by Q upon epsilon naught. So Q is flux into epsilon naught. They are giving the flux directly. Just multiply by epsilon naught. 1.05 into, we know the value of epsilon naught, which is 8. It is like what? 8.8 into 10 to the power minus 12. So yeah, just multiply these two, you'll get the answer. So you should get option A. So it is 1.0 into epsilon naught. I remember it is 8. Exact value I'm not able to remember, but if you multiply these two, you'll get slightly more than 9. Okay. Next one. So this is also simple. Basically, they are saying if this is plus Q, what will be the charge density here? So guys, we know this is a metal. So on the metal, the electric field will be 0. So on this surface, if we talk about the electric field due to, let's say the charge for this one is, let's say charge is Q. So KQ divided by R1 square. This field must be equal to the field that is created by this. Equal and opposite. So it is equals to the field created by this. So guys, the field created by this small q. Okay, let's use different symbol for the charge on this one. So okay, let me explain you what we are doing. So the charge here is plus q. Okay, so it will create electric field, right, which will go like this. But inside there must not be any electric field. That is the property of a conductor. So on this surface will have a negative charge density, let's say minus q. Why? Because this minus q will have electric field in the opposite direction. Okay, so this basically minus q, what will be the electric field? So inside, of course, due to this minus q, there will not be any electric field. But if you talk about outside this area, electric field is in this direction. So this will cancel the electric field which is produced by this. So if we talk about inside, guys, what is the magnitude of magnetic field which will be created? So as we know, magnetic field created is given by, they are asking the surface charge density. So if we talk about the magnetic field which is created, so we know it is kq by r square. So this is the magnetic field due to point particle, kq by r1 square, and that must be equal to the field created by this one, the charge at minus q. So guys, we know the field created by a sphere with, let's say, the surface charge density is sigma. We know it is going to be sigma by 2 epsilon naught. Similarly, the field will be created by this one also. So we can write that the field created will be sigma by epsilon naught. So this we can use to find the sigma, which is what they are asking, surface charge density. We have to put minus sign because it is opposite. So now if we solve this, sigma will be minus epsilon naught k. K is 1 by 4 pi epsilon naught, so epsilon naught cancel, 1 by 4 pi q divided by r1 square. That is our answer. So important thing to remember was the electric field due to sphere if the charge is sigma. So this is the electric field, and this is all due to the charge on this surface and as well as on the outer surface. Alright, let's continue. Next one, so this is kind of simple. They're asking particle charge is given thrown. Let's say it is thrown like this. Electric field is there against, so electric field is in this direction, velocity is in this direction. What is the distance it will travel before it stops? So there are two ways to do it. One, we can use potential energy, kinetic energy, energy conservation, or we can find force and then find acceleration and then use equation of motion. So I think energy one is slightly easier. So finally it stops, so energy becomes 0, but there will be potential energy. So guys, all the kinetic energy is converted to potential energy. So what is the kinetic energy? Half mv square. That is converted to potential energy. Potential energy is charge into the potential difference, and here that is going to be electric field into the distance traveled, which is, let's say, d. So we can find d. We know everything, half mv square qe. We can find the d. So simple, right? Energy, if we go by the energy one. Okay, new for this is Coulomb's law, guys. This is also simple. Okay, so what we have to do is 2-point charges are given. So first we need to apply Coulomb's law. K q1 q2 by r square, that is what, 10. Okay, now what we'll do is we'll do q1 is q1 minus 6, because what we are doing is additional charge of minus 6 micro. So q1 will become q1 minus 6 and q2 will also be q2 minus 6. So we'll get another equation and we'll get the values. Alright, so here if I write the equation again, so this is k q1 q2. What is q1? It is 3, it is 4, and now if we put additional charge, so minus 6, so this will become minus 3 and this will become minus 2 divided by r square. So guys, as you can see, this will become half because it is 3 into 4, it is 3 into 2. So it will become half, so answer is 5. So this was very simple. Okay, this is again very simple. Object has this much atom, electron is removed. What is the charge gained? By 1 gram. So guys, we know in 1 gram of 1 gram how many atoms will be there. So 1 kg is 1000 grams, so in 1 gram 4 into 10 power 17 atom. And if we remove 1 electron, so how many electrons we have? 4 into 10 power 17. What is the charge on each electron? 1.6 into 10 power minus 19. So this is your answer. So solve this one and you will get the answer, which will be option B. Alright. Okay, steady state they are asking. So guys, in steady state we know there will be no current across the capacitor. So there is no current here. The current here is 0. So it means the potential, this we can take it as open wire. Alright, so this is basically the current is not flowing. So whatever is the potential difference, let's say this is A, this is B. So whatever is the potential difference between A and B, that is the potential difference across the capacitor, and they are asking the charge, so potential difference into the 4, that will give you the answer. So how do we calculate VA minus VB? Very simple. So here it is internal resistance also. So if we draw the diagram, it is, so this is the diagram. So this is 2.5, this is 0.5 ohm, this is 2 ohm. That's it. So guys, this is the diagram. We need to find potential difference across these 2 points, A and B. So I believe this is very simple. 2.5 is there, so, and resistance is also 2.5. So potential difference between A and B will be 2. So 2 into 4, that will be 8. That will be the answer, 8 micro coulomb. Because this is actually 4 micro, right? So that is why. Okay, next one, resultant. So this is all about drawing it correctly and you'll get the answer. So guys, this is, if this is A, then this is also A because they are connected. And let's say this is B, then this is also B. So if you just look at between A and B, this is 20 ohm. So let me connect A and B. So this is 10 ohm directly. Then this side we have 20 ohm. And this side also between A and B, if you see, this is 20 ohm. So this is also 20 ohm. So this 20, 10 and 20, they are in parallel. 20 and 20 is 10, 10 and 10 is 5. So the resultant is 5. Very simple. Okay. contact. So this is basic knowledge of PN junction, guys. When we increase the current, the contact potential that will not remain changed. The contact potential is not related to the current in the PN junction. So this is basically from basic knowledge of PN junction. Okay, so kind of memory based. Okay, next one, reverse bias. So guys, what is a reverse biased? So guys, in reverse biased, we know that, okay, so if I draw the PN junction, this is the symbol. So guys, in there are two kinds of bias, we know, forward and reverse. Okay, so in reverse and forward, what is the difference? The difference is about the potential difference. So if we talk about this one, let's say A, so we can see that this is at lower potential, this is at higher potential. So basically this VA and VB. So this is the how we connect it. So if VA is greater than VB, that is forward biased, and vice versa. If VB is greater than VA, that is reverse biased. So all we need to see was in which case VB is greater than VA. What is VB? Which is connected to this base of the triangle, and VB is connected to the vertical line. So here guys, as we can see, if VB is greater, that will be the answer. So here we can see vertical line, this is 5 volt, this is earth, so this is 0. So vertical line is at higher potential. So second one is basically the reverse biased. What about the other one? So here A, this is -5, this is -12. So it means vertical line is at lower potential, right? -12. Okay, you can take any 4. This is 5, this is 10. So again, the base of the triangle is at higher potential, but it must be lower. So basically, this is what you need to know, and then you can see the option, and then you'll get the answer. Okay. Very simple, following circuit logic function. So we can just draw truth table. So let's say if A is 0, B is also 0, what will that be? So this is NOR gate. So A and B first OR. So OR will be 0 and then NOT. So it will be 1. So this will be 1 and 1. And this is again, guys, it is a NOR gate. So 1 and 1 is 1, but NOT will be 0. So if A and B are 0, if I draw the truth table for this. So A and B, and let's say the resultant. So if this is 0, this is 0, then this I am also getting 0. So similarly, I need to do, let's say 1, let me take 1 and 1. So if this is 1 and this is 1, so OR will be 1. So this will be 0, so this is also 0, 0 and 0 is again 0, but finally we'll get NOT, so it is 1. So if this is 1, 1, then I get 1. So similarly, you can draw for other also. Okay, but we can compare using this. So if you draw the complete table, this will be AND gate, which is also visible now. Other can be eliminated, it is not NAND, it is not. Okay, OR is also possible using this, so we need to do the 1 0 1 also. So if we do 1 0, we'll get 0. Alright, so that is the thing. Why is that 1 0? So this will be 0. So this is OR. Yeah, so this is actually AND gate, right? So 1 and 0 will be 0 and NOT. So this will be 1, 1 and 1, again 1, then again 0. So 1 and 0 will give us 0. So this kind of behaves like AND gates, right? This is what? This is NAND gate, not AND, right? So using this knowledge we can. So this is AND. What is the symbol of OR gate? Okay, we'll see that in the last question. But let's solve this on transistor. So very simple. We talking about collector current. Remember, collector current is alpha times the emitter current, IE. So everything is given. They are talking about the change. So delta formula will be same. Delta IC is alpha into delta IE. So alpha is given, right? Delta IE, which is the change in the emitter current, that is also given. What is that? 100 milliampere. So alpha into 100 milliampere, that will give you the answer, which will be 95 milliampere. Okay, 95 milliampere. Okay, last one. So these you should know, guys. This is memory based. What is the symbol of each? So this is NOT, and then this small circle is basically NOT. So this is NAND. Okay, and this is the symbol of OR gate, right? This is OR gate. But then we have this. So this is NOT, and then we have this also. So this is basically the symbol of exclusive OR gate. Okay, XOR, which we call. Okay, then we have the C, which is NOR gate, and then we have this is NOT gate. So they are asking XOR, which is basically this exclusive OR, and that is basically in this diagram, and then asking NOR. So this is OR and then NOT sign. So C1 is the NOR. So we'll compare and we can see that the first one is B, second one is C. Alright guys, so this was the last question, kind of simple one. But okay, there was some question which were a little bit difficult also. So yeah, this is it. This was mega test 1, complete syllabus of 12th class. In the next mega test, we'll have complete syllabus of 11th class. Alright, and I wish you luck for mega test 2. Be prepared, and thank you very much for attending this, and have a great day.